full of success. A very good morning to all. It is a true privilege to welcome you to this year's National Conference on Innovations and Emerging Trends in Natural Therapeutics, Molecular Biology and Structural Analysis of Biomolecules, IETNMS 2024. A very good morning to one and all gathered here. Today, we are surrounded by some of the brightest minds and visionaries in this field whose groundbreaking work influences our understanding of life itself. This conference represents an incredible opportunity for thought-provoking discussions, innovative research, and collaboration towards addressing future challenges.
God speaks in the silence of the heart. Listening is the beginning of prayer. Let's start our day with prayer and end it with a gratitude. Let us start this auspicious day with a prayer. I now invite Kavya to present the prayer song.
கைத்தொழவே எதுவும் கைகூடும் தும்பி கையனை மெய்யனை கரிமுக வரதனை கைத்தொழவே எதுவும் கைகூடும் செய்தொழில் சிறப்புற செய்தொழில் சிறப்புற சிந்தை தெளிவு பெற வைத்திடுவோனை வேலமுகத்தோனை வைத்திடுவோனை வேலமுகத்தோனை கைத்தொழவே எதுவும் கைகூடும்.
Thank you, Kavya, for that soul-stirring invocation. May this day be filled with fruitful learning and inspiration. Now, I would like to welcome all the dignitaries on the dais to light the lamp.
A true leader is one who knows the way, goes the way, and shows the way. It's my pleasure to invite Dr. D. Victor Arokya Das, sir, Associate Professor and Head, Department of Biochemistry, PSGCAS, to deliver the welcome address.
Good morning, everyone who are present online and offline. I would like to welcome everyone gathered here. There are around 200 participants who are on the online mode also. At the outset, I would like to welcome our Secretary Sir in absentia, Dr. T. Kannai, who has to go for an urgent meeting to the PhD Tech. I would like to welcome our beloved Principal in Charge, Dr. M. Sengutuvan, a guiding force in the college. I would like to invite the resource persons who have come from various parts of the country and also from the other country like Singapore. I would like to welcome Professor Kini RM, Emeritus Professor, National Institute of Singapore. I would like to welcome Dr. Professor Karnagaran. He was our own alumni, faculty here, and then he has gone to great heights, and he has come back as a great scientist. Welcome you, sir. And I would like to welcome my beloved long-time friend and industrialist, a great scientist, Dr. Lakshmi Subramaniam. Welcome you, sir. I would like to welcome Dr. Sridevi in absentia. She started from Madurai Kamaraj University. She'll be joining us very shortly. She's our own alumnus, a great alumnus and professor of molecular biology, plant molecular biology at Madurai Kamaraj University. And I would like to welcome all the organizers, our own department faculty, the dean of various departments, the HODs of various departments, staff members, as well as the staff members who are online participating and presenting papers in the day-long event. And I welcome each and every participant and also the other press members and everyone who are assembled here. I wish you a great event, the whole day-long event to be a success. Thank you.
Thank you, sir, for your warm welcoming words.
Now, I would like to welcome our Principal in Charge, sir, to honor our Chief Guest, Professor Kini RM, Emeritus Professor, Department of Biological Sciences, National University of Singapore. I would like to welcome Dr. D. Victor Auroquedas, sir, Associate Professor and Head, Department of Biochemistry, PSGCAS, to honor Professor Devarajan Karnagaran, sir, Emeritus Professor, Department of Biotechnology, Chennai. I would like to welcome Dr. B. Ramesh, sir, Associate Professor, Department of Biochemistry, PSGCAS, to honor Dr. Lakshmi Subramanian, sir, Chief Scientific Officer, Swasti Veda Private Limited, Mumbai.
The heart of leadership is about understanding people, inspiring them, and empowering them to reach their full potential. It is with great pleasure and excitement that I extend a warm welcome to our esteemed Principal in Charge, Dr. M. Sengutuvan, sir, Associate Professor and Head PSGCAS. We are truly honored to have you with us today. Your vision has always been a guiding force. I now invite you to deliver the presidential address to this esteemed gathering.
most respected Secretary of this esteemed institution, Dr. T. Kannayan, sir, respected my dear brother, Dr. Victor Arokia, sir, head of the Department of Biochemistry, esteemed invited speakers from India and abroad, respected and my beloved brother, Dr. B. Ramesh, Associate Professor of Biochemistry Department, my teaching fraternity of Biochemistry Department, heads of various departments of our college who have come over here to participate in this great program, teaching fraternity from other departments who have come over here to participate in this program, a highly knowledgeable, renowned delegates from our institution and other institutions of India and abroad, my dear student friends, I am very happy to be here in this morning after attending the program in social work, that I feel that I am coming from society to college. So social work is very much associated with society for doing all social work. So, but this is very important, a product from the department going to society. That too with a very innovative method that you people have to do, and that is also ethically binding to IPR for the betterment of human society at large. So it gives me immense pleasure to welcome you all to this one-day national level conference on innovations and emerging trend in natural therapeutics, nanobiology, molecular biology, structural analysis, and biomolecules, hosted by the Department of Biochemistry of our college. Because innovation and emerging trend is very important nowadays. To go, whichever may be the field, innovative thought is very important. That too in a holistic manner. So this event brings together an exceptional assembly of minds, students, researchers, and faculty united by shared passion for life sciences and its boundless potential to transform human health and well-being. So as the life science students, you are all the forefront of a revolution in understanding the intricacies of the living system. This conference is an opportunity to explore how cutting-edge technology or disciplines such as natural therapeutics, nanobiotechnology or nanobiology, molecular biology, they converge to unravel the mysteries of life, diseases, and innovative sustainable solutions for global challenges. So today's theme, which is focusing on natural therapeutics, which emphasizes the timeless connections between nature and medicine. So it is very integrated one. We cannot, it is a separate, it's not a separate entity. Nature and medicine. We take materials as a medicine, we find some materials as a medicine from nature only. So from traditional herbal remedies to the identification of potent phytochemicals, natural compounds continue to inspire modern drug discovery. So you may understand this point because how the antibiotics structures have changed from time to time to fulfill the today's requirement, because all the microorganisms have mutated enormously, that you know that. So these bioactive molecules hold the promise to combat antibiotic resistance, and they target complex diseases and enhance overall health. So exploring this frontier demands an interdisciplinary approach combining the knowledge of botany, phytochemistry, pharmacology, and biochemistry. And similarly, the field of nanobiology is revolutionizing the way we diagnose and treat the diseases. By harnessing the power of nanotechnology, researchers are developing drug delivery systems that are more precise, efficient, and less invasive. So the synergy between nanotechnology and molecular biology is opening pathways for targeted therapies that can potentially cure once incurable conditions like cancer or neurodegenerative diseases. So the third focusing area, molecular biology and structural analysis of biomolecules, forms the foundation of life sciences. Understanding the structure, function relationships of protein, DNA, and RNA is pivotal for innovations in personalized medicine, vaccine development, and biotechnology. So structural biology techniques like X-ray crystallography, NMR technique, cryo-electron microscopy continue to shed light on the molecular underpinnings of life and have been instrumental in breakthroughs like mRNA vaccines. So this conference offers an enriching platform to delve on or into these fascinating domains, and I encourage all of you to actively participate, ask questions, and engage with our esteemed speakers and experts. So today's sessions, encompassing keynote addresses, poster, and paper presentations and discussions, are tailored to inspire innovation, spark curiosity, and foster collaborations among the bright minds like you people. So I extend my heartfelt gratitude to the organizing committee for their tireless efforts in bringing this event for fruition. My special thanks to the distinguished speakers and guests for sharing their expertise and insights. So your presence adds immense value to this event, and we are deeply honored to have you with us. So to our students, I urge you to take full advantage of this platform, make use ethically, morally, scientifically, in a knowledgeable way. This conference is not just about acquiring knowledge, but also about envisioning how you can contribute to the science and society through your research and ideas. Remember, the future of life sciences depends on inquisitive minds like yours, ready to challenge norms and embrace innovation. Let us begin this day of intellectual exploration and collaboration with enthusiasm and a commitment to advance the frontiers of life sciences. And I am very much associated with this department for the past 31.5 years of my service here, right from January 11th, 1993. I took general biology and biology practicals for about 15 batches from 93. So I was very happy, and whenever I enter into the biochemistry department, I feel like that I'm in a family looking my child, because biochemistry is a bond between a marriage happened between biology and chemistry. So biochemistry. So it's a child, a very good growing child. You see, dynamic people are here. I know that. I feel very happy to be here, and I wish you all success and inspiring the conference. Thank you very much.
Thank you, sir, for your enlightening and motivating words.
As we embark on IETNMS 2024, we celebrate both our accomplishments and the opportunities ahead. This conference is a platform where ideas converge to promote human well-being and global growth. It is a great pleasure to invite Professor Kini or Manjunatha, Emeritus Professor, Department of Biological Sciences, National University of Singapore. He was born in Shimoga, Karnataka, India. He did his undergraduation in University of Mysore in 1977 and ranked 47th among 15,000 BSc graduates in the university. Then he completed his master's in biochemistry in the same university in 1979, ranked first in MSc Biochemistry in the university. He did his doctorate in University of Mysore in 1983. He is having citations of 20,590 and H index of 79. His research areas are protein chemistry, structure-function relationships, protein-protein interaction, and protein design and engineering. He conducts basic and applied research on snake venoms and saliva of blood-feeding animals. Both contains pharmacologically active proteins. He is an accomplished biochemist and established world leader in the field of toxins as well as in blood clotting. His main research focuses from toxins to therapeutics, where he identifies novel bioactive proteins from venoms or saliva, determines their mode of action, and designs potential drug leads based on their structure. Venom toxins target cardiovascular and neuromuscular systems. He has published 240 research publications, including 182 original articles, 40 reviews, and 18 book chapters. In addition, he also filed 60 patent applications, including 34 issued patents. He edited 2 monographs, Venom phospholipase A2 enzymes, structure, function, and mechanism, and toxins and hemostasis from bench to bedside. He also edited 5 special issues for various journals. He was the chairman of a subcommittee of the Scientific Standardization Committee of the International Society of Thrombosis and Hemostasis from 1998 to 2016. He was a member of the Council of International Society on Toxinology for 12 years and the president of the society from 2019 to 2020. He was the vice president of the International Proteolysis Society for 2 terms. You inspire us through your achievements, sir. It is my honor to welcome you to deliver the inaugural address.
Good morning. Today I see a lot of young faces, so I would like to just give you some idea how you should chart out your future. So first, obviously, you need to start identifying where you want to see yourself about 15, 20 years from now. So you need to start thinking right now so that you know where you would like to go. It is very important to identify your goals in life, what kind of position you would like to occupy. This is important mainly because you need to have a destination. Imagine you're going to a bus stop. You're not going to take the first bus which comes in. You wait for your bus which takes to your destination. If not, you'll never reach your home. So it's very important to recognize that. So therefore, you need to start identify your destination, and then you know which bus to take, which train to take, where do I need to go, what do I need to do, what are all the things I need to collect. So it is very important to start recognizing your destination and the right buses, right vehicles which take you there. To do that, obviously, you are young, you are going to start understanding various areas, not just focus on one area and say, I found something. So it is important to understand because you are going to be in that field for probably about 40 to 50 years or maybe more by the time you guys reach retirement age. The retirement age may be 70 or 75. So you need to really have a long way to go and you need to enjoy what you are doing. So you have to prepare kind of strategies. How do I go about it? How do I achieve what I want to do? Of course, life is not written in a very clear path. Just because you want to become something, You are going to have a lot of obstacles, a lot of competition to face. All that is fine, but you are going to, if you don't have an objective, you don't have a destination, you will never reach there, okay? So there are a lot of opportunities, really a lot of opportunities. Most of you are undergraduate and master students. You have a huge place open for you. Of course, that's one step you have already achieved. You're all PSG colleges students. You have a very proud foundation. You have wonderful people who have graduated and fantastic alumni over last several decades. So you have already laid the foundation. Use that foundation, and you also have a lot of network. People are in very good places, so you need to meet with them, talk to them, and find out, and link with them, and that's how you create an alumni network of your own, so that you know how to go. Of course, as you go up, you also get the next generation of people with you. The second thing you need to do is you need to, whatever you achieve, you would like to do the first time. How many of you remember who landed on the moon first? By show of hands? A little bit higher. I can't even see from here, yeah. A lot of you know, right? Do you know who came in, who landed second? So there's only one hand. And do you know there has been 6 lunar efforts to land, and 12 people walked on moon. Do you know who was 3rd, 4th, 5th, 6th? Nobody knows. So you have to be the first one to land. So whatever you do, you have to be the first one in the world to do things. When you do that, everybody will remember, okay? So if you imagine, who was the fastest sprinter? Do you know? Usain Bolt, right? Nobody knows who came in second. Same with everything you look, every field, you need to be the first one to reach there, okay? It is important. It's not that hard. All you have to do is think what you want to do, and you are carving out a niche area, and much easier for you to start thinking, that's what I would like to do, okay? So it is important to be the first one to be there. Of course, to do that, you need to be confident about yourself, right? So how do you become confident? You have to recognize your successes in life. I'll tell you, how many of you remember when you were in the 4th grade that mathematics was very difficult? When you started, you always told your classmates, this year the decimal points are difficult, division is difficult. 5th grade, 4th grade looks nothing. It's the algebra, right? And when when you started in the 5th grade, you said, it's very difficult. I was able to count like this, but now a squared by b squared, you cannot use your fingers and toes anymore, right? But 6th grade, algebra is nothing. Trigonometry is impossible. Like that at every stage, whether it's math or physics. In physics, you learned magnetism, light, sound, electricity, all kinds of things. So similarly, every year, I'm sure you have talked to your classmates, it's impossible when you started, and by the end of that year, you're not bad, you managed doing that, and you're familiar with it. So this happens all the time to every one of us. That happens because all of us start from 0. Okay? By the end of your high school, you would have finished at least about 800 to 1,000 subjects, all starting from 0. And by the time you finish, you mastered all those 800 or 1,000 subjects. By the time you finish your pre-university or junior college, you would have gone to about 1,500 subjects. By the time you finish your undergraduate about 2,000, 2,500 subjects. You have studied 2,500 subjects, all from 0, and achieved what you want to achieve. 2,501 should not affect you. So you should be thinking that I can do that, because only one and the second one is a little bit difficult. But when you have done thousands of subjects, next one should not be scary. All that I'm trying to tell you is you are capable of learning. This you have done it many, 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 many times, thousands of times, not 3 times, 4 times. So when you put that information in your mind, you'll see everything looks simple. It's not complicated. Because when you have done 1,000 times, 1,000 first time, it shouldn't make any difference. It will take some time, yes. You also start from 0, yes. But you can do that. So that's one thing you need to put in your mind. Once you start doing that, because these are all your accomplishments in last 15 years of 10, 12 years, 15 years of your learning. You have accomplished so many things. So you have to remember that. That means you are all capable of learning. Okay? And once you remember that, everything becomes easy. Of course, in life, you have also failed. Everyone has failed. When you got up when you were a kid, you fell down before you started walking. Everybody did. Nobody walked on the day 1, right? So everybody had failures. You try to understand, learn lessons from the failures, and keep going forward. That's all you need to do. And you have to enhance your horizon, your ability to see things. So to do that, you have to read a lot. All the information you get your hands on, please learn, please read. You need to understand what's going on in your contemporary science. That means it's independent of whether it's physics, chemistry, math, biology, biochemistry, molecular biology, nanotechnology, it doesn't matter. You need to understand what's going on at your time, everywhere. So you have to at least keep an eye on what comes out every week, every month. For that, there are various ways of strategies to do that. You should do that. Read everything. And you also have to develop network. Okay? As you grow, you have to develop network and develop your ability to see different things. This you can achieve by going to different labs. Say, imagine you have worked for your MSc project or BSc project in one lab, go to another lab to do MSc project. Maybe go to a different institution. I don't know how it is in PSG. It is important for you to go to a different place. Not that other place does better. It is important to understand everybody has a perspective. The way we look at things are different from each of us. So that's what you have to improve. So you need to learn to understand information from different viewpoints. And when you go meet with different people, of course, after MSc, for PhD, go to a different institution. After you finish PhD, go to do postdoctoral or whatever. Whatever field you are, try to do it in different places. I'm telling you only from point of studies, but it's the same thing if you want to become a lawyer, you want to become a CEO or something. You have to go out and do those things. And as I told you, nothing is difficult because you've already proven yourself thousands of times, not once or twice. So you have to put that thought in your mind, I'm capable of learning. And you have done that so many times, so many times. So it should not harm you from that, okay? And of course, when you do at different places, every place gives you a different network, different connection, different perspective. It helps you to improve your horizon. You can see many things which you are not seeing from the same place. So you're moving to different places to see things. It is very important for you to do. And of course, please listen to seminars, not because I'm giving you one today, but it's very important for you to learn from different people. The reason why it's important is you're getting a lot of information, cutting edge information, within about 30, 40 minutes or 1 hour, whatever the length of the seminar is. So you'll get really cutting edge information in that field. It's different field, but it gives you a different perspective. It helps you to understand something new, something different. It helps you to kind of think how to manage your strategies, okay? And finally, of course, try to attend conferences. It also helps you to learn from different people. It's important not just go to the conference, meet people, talk to them. Nobody is going to bite you. You have to talk to them, understand. If somebody acts very funny, in the sense he knows a lot, then it's his loss or her loss. So you have to understand, when you have a question, go ahead and ask it. Don't worry about what others think. There are no stupid questions, but there are stupid answers, okay? So with this, I would like to stop my inaugural address just to help you to understand where you can go. Sky is the limit, and what you want to do, sky is the limit. Whatever you want to do, you can achieve. I'm not saying this, but I'm telling you, all you have to do is keep in your mind that you are capable of learning. Once you put that information into your hearts, nothing is going to stop you, okay? Good luck. Thank you very much.
Thank you, sir, for your thought-provoking words and sharing inspiring stories to illustrate the potential for success.
Gratitude is the memory of the heart. To conclude this inaugural segment, I would like to invite Dr. B. Ramesh, Associate Professor, Department of Biochemistry, PSGCAS, to deliver the vote of thanks.
Developing the attitude of gratitude sweetens even the smallest moments that worth more. A wonderful morning to each one of you present here. I am deeply grateful to our esteemed management for their invaluable guidance and steadfast support in conducting this national conference. A heartfelt note of thank you to Dr. T. Kannayan, our dedicated secretary in absentia. I express our sincere appreciation to Dr. M. Sengutuvan, principal in charge, for being a guiding force in our institution and in conducting this conference. My gratitude extends to Dr. D. Brinda, former principal as well as HOD of the Department of Biochemistry, for her unwavering commitment to our institution. I take this opportunity to express my sincere gratitude to the guest speakers for the day, Professor Kini R.M., Emeritus Professor, Department of Biological Sciences, National University of Singapore, Professor Devarajan Karunagaran, Emeritus Professor, Department of Biotechnology, IIT Madras, Dr. Lakshmi Subramanian, Chief Scientific Officer, Swasti Veda Private Limited, Mumbai, Mentor, Center of Excellence in Medical Textiles of Citra, Coimbatore, and Dr. G. Sridevi, Associate Professor, Department of Plant Biotechnology, School of Biotechnology, Madurai Kamaraj University, Madurai, for accepting our invitation and making time for this conference. A special thanks to Professor Kini for enlightening the young minds in the inaugural address. Thank you, sir. My heartfelt gratitude to Vice Principals for walking us along in every venture taken up. I express our deep sense of respect and regard to Dr. D. Victor Arogedas, Head, Department of Biochemistry, who is guiding light for not just this event, but for any initiative. Thank you, sir. My wholehearted gratitude to all the staff members of the Department of Biochemistry, Microbiology, Biotechnology, Nutrition, Botany, and Zoology, who have always been a source of great strength and constant channels of input to all the events undertaken by us. Warmest gratitude to all the participants, both offline as well as online, for your commitment towards this conference to make this a grand success. A special appreciation to the students of the Department of Biochemistry, Microbiology, Biotechnology, Nutrition, and Botany, as well as Zoology, for your commitment towards the conduct of this conference. Hope today all the sessions will give all of us an opportunity to embrace changes for the better. Thank you all once again.
Thank you, sir. Let us engage, learn, and grow together as we navigate through today's sessions.
A good teacher is like a candle. It consumes itself to light the way for others. Let us begin the first session. I like to invite Professor Kini Aram, Emeritus Professor, Department of Biological Sciences, National University of Singapore. The stage is yours, sir.
Announcements. Participants of oral offline presentation kindly assemble at the Sangamam Hall. I repeat once again. Participants of oral offline presentation kindly assemble at the Sangamam Hall immediately.
Can you hear me in the back? Can you hear me in the back? Wonderful, thank you. So today I'm going to talk to you about atrial peptide analogs for tailored treatment of acute decompensated heart failure. Okay? So as I was introduced that I work on venoms. I work on venoms of many different animals. Of course, snakes, that's our main focus. Over the years, we have also worked on scorpion venoms, spider venoms, honeybee venoms, ant venoms, conus venoms, sea anemone venoms, and many other species of animal venoms. Okay? Our interest is to study toxins, their structure-function relationship, and mechanism of action. You have also heard that we work from designing drugs from toxins to therapeutics. Okay? And of course, when you want to develop new drug molecules, you have to isolate and study new toxins. We always work, my lab works only exclusively on new toxins. Okay? And about 15 years ago, on animal venoms I've been working for close to about 45 years. Okay? And about 20 years ago, we started working also on blood-feeding animals, such as ticks, mosquitoes, and their saliva. Because the saliva of these animals also have very potent bioactive molecules. You also know these blood-feeding animals actually transmit various infectious diseases, malaria, dengue, chikungunya, and so on and so forth. So these pathogens, if you purify them and inject, their infectivity is very, very low. But if you mix it with relevant saliva of the vector which transmits it, then the infectivity jumps up by 3,000 times, 5,000 times, 10,000 times. So something in the saliva of these animals enhance the transmission of these pathogens. Because of that, a single mosquito bite is sufficient to cause malaria, dengue, and chikungunya. or any of these diseases. If it was not from the mosquito, I have to inject 5,000 times those malaria parasite to get you malaria. Very difficult, right? So that's the advantage of it. So we study these also. So we study venoms because you get toxins which target neuromuscular junctions, neuromuscular systems, cardiovascular system, and channelopathies. Channelopathies means the ion channel drugs. There are so many diseases caused because of either some ion channel is very active or not so active, so you can control them. We work on these blood feeding animals because they are potent sources of anticoagulants, antiplatelets, vasodilators, immune suppressors, as I kind of told you, transmission enhancers, transmission of the pathogen, okay? And of course, we have so many species. Of course, I just put few species on the picture. You can imagine how many species I could work on. Can I get water, please? Okay, yeah. So that's what it is, okay? So how do we go about it? You need to understand, just because you have a mosquito or a tick or the venom, you can't do those things, right? So for example, whether it's or other venoms or saliva, they're all mixture of 30 to 50 molecules to hundreds of molecules, some close to about 1,000 molecules, okay? So when you have, like some of the snake venoms, you can get in large quantities. For example, king cobra, if you milk, you'll get about 1 gram, 800 milligram of venom. So when you have a lot of it. But cobra, common cobra, Indian cobra, about 150 milligrams. And crate gives you only 15, 20 milligrams. And there are other venoms, you get only 2 milligram, 5 milligram. And of course, you can imagine mosquito saliva, you'll get few micrograms, few nanograms of it, right? So you, based on what you have, how much you have, if you have large amount of it, like snake venom, which is very good, you can fractionate them by chromatography. You understand all these chromatography, right? So you can fractionate them and isolate the molecule you want. So that's how you get the new toxin, okay? But when there is very small amount, like mosquito saliva, tick saliva, you have really pathetic amounts, right? So what we do is we use proteomics. Proteomics, I am sure many of you have heard of it. Has it been taught? Yeah. So we use proteomics. Of course, we can also do cDNA library of the venom gland or the salivary gland. All the molecular biology students are already familiar with it. So we do cDNA library. We also do transcriptomes and genome by next-gen sequencing. You're also familiar with Illumina sequencing, I'm sure, right? Yeah, 10x technology. There are many RNA-seq, many things, right? So we do those. And when you come from proteomics, cDNA library or transcriptome and genomes, you just have a new sequence, new protein. You have identified a new protein. You don't have the protein in your hand. When you come here, of course you have protein in your hand. You have a physical molecule. Here you need to make this. So if the molecule is very small, we use peptide synthesis. By chemistry, we make the peptide, okay? And if it is very large protein, we do the cloning and expression. We use E. coli, we use yeast, we use insect cell line, we use mammalian cell line. Based on our needs, we use all of these things and create the molecule, make the molecule. And of course, sometimes you also need to fold these molecules to have the natural. So once you complete that, now you have the molecule in hand. This is our first step. Once you have this molecule, we study its structure, its function, and mechanism. We do its structure by structural biology. We do NMR, X-ray crystallography, cryo-EM. Today I'm going to talk to you a little bit about cryo-EM. Not much details, just to show you a little bit of it, okay? We want to know the function. I told you it's a new protein, new toxin. We don't know what it does. So what we do is we inject into mouse. We do 1 or 2 mice to find out what it does. And based on the behavior, we identify, oh, it shows effects on the heart. So it causes neurotoxicity. It does something for its movement or something. So we understand that. Then we go to the tissue, organs and the tissue, and isolated tissue we study, and then study the mechanism. We want to understand exactly where does it go and bind, how does it cause this function. So basically we do protein chemistry, proteomics, molecular biology, transcriptome, peptide synthesis, cloning, pharmacology, and biophysics. Okay? Once you complete all of this, we know it's the target. It could be a receptor, an enzyme, or an ion channel, right, to which the toxin would go and bind. And we all know entire toxin need not go and bind there. You need only a small portion on the surface of the toxin is responsible. So we want to identify that site. So that's the functional site of the toxin. We use structure-function relationships. So for this, you are also familiar, you can do site-directed mutagenesis, deletion, and omission peptides and things like that. So many things you can do sorry, to identify the functional site. Of course, this functional site is sufficient to go and bind to the target protein and show its function. We also know sometimes when you isolate the functional site separately, it may not retain the same conformation as the native protein, right? So what we do in such a case, we take that functional site and put it onto a molecular scaffold. These are mini proteins. We have a lot of disulfide bonded mini proteins with different conformation and structure. So we put the conformation we need and use protein design and engineering and put that functional site on top of this molecular scaffold so that it retains the same conformation or similar conformation as the native protein. So now this scaffold can go with this functional site and recognize the target and do its job, right? The reason why we do that is sometimes toxins have a toxic site and a beneficial site. We don't want the toxic site. So when you use this technology, you the toxic site, okay? And you have a molecule which has pure beneficial side of it. Is it clear to everybody? So this is how we design and develop a therapeutic agent. So there are several examples in our lab. This is a small list just to give you a flavor. So this is toxin isolated from mamba snake venom. And this toxin, calciceptin, binds selectively to L-type calcium channel. It does not have N-type, T-type, P-type, Q-type calcium channel, okay? And we showed that is red colored segment is the functional site of this toxin. 8 amino acid residue peptide designed based on this bind selectively to L-type calcium channel. It does not touch N-type, T-type, P-type, Q-type channels. When you block the L-type calcium channel, the blood pressure drops. Okay? So based on that, we have designed hypertensive peptide. Based on these green segments, we have designed antiplatelet peptides. Based on this white segment, we have designed anticoagulant peptides. This is from king cobra snake. And based on this segment, we have designed an analgesic peptide. This peptide is 20 to 200 times more potent than morphine in various pain models of animals. It is orally active, sublingually active. You can also inject, and it works from many different routes of administration. It's good for acute and chronic pain, okay? So, but when we finished all the experiments at a company called Therapeutics, and we finished all the animal experiments, when we were just looking for money to take it to preclinical toxicology studies and phase 1, phase 2 studies, 2008 financial crisis hit us. So when 2008 came, all the money disappeared from the market because all the VCs went into shell. So that's a different problem, but basically we do from toxins to therapeutics, okay? We also do how to develop them to be important, okay? I kind of minimization. So from big protein, we identify the segment we need, and that's how it is. Then, of course, you need to improve its potency. We also do some tricks on it to make it a lot more active, biologically active. And, sorry. And we also develop them for oral availability, sublingual availability, because you can kind of develop them for easy compliance, okay? And we can also increase the half-life of molecules so that they stay in blood. You don't have to take it every 2 minutes, okay? Slow release formulations, okay? And with this, I want you to understand, we could do, because we start from novel toxins and go through all of these things, you also learn a lot of things in basic sciences, because you start understanding many things. You can characterize new toxins. We also try to understand, how do these new molecules come from? Where do they come from? You know, nothing has come from. You have to understand how they are. And we study a lot of these different things. I'm not going to go through all of this. So every one of them, we learn a lot of new lessons, very exciting things. We also have something in both basic and applied. They kind of straddle between these two. For example, common cytolytic regions, it is important to design antimicrobial peptide, anti-cancer peptide, and things like that. And common structural feature flanking segments of protein-protein interaction site. We could identify interaction site directly from the amino acid sequence. So things like that. We also have contributed to structural determinants in protein folding, how the disulfide bonds connect. When you have multiple disulfide bonds, how do they connect to the proper cysteine? You need to understand those things. And we have developed drug delivery systems, universal method for fishing, orphan receptors and ligands. So these are both basic and applied. You have a lot of applications, okay? And we also have many. I've just given you some list of it. So I talked a little bit about analgesic peptide. This molecule, angiostatin molecule, was good for diabetes, retinopathy, and macular degeneration. And inhibitors, it's in preclinical toxicology studies now. So like that, there are many. Today I'm going to talk to you about this, personalized treatment of heart failure of patients, okay? So let's look at the people who can't. This is Sindhuja, my PhD student who started this project, and Mark Richards, Miriam Rademacher. These are from, he's the Cardiovascular Research Institute director. She's from Christchurch Heart Institute in New Zealand. This is Professor Ogawa and his students from Japan, Kyoto University. And this is my PhD student. He went and studied in Andreas Patzak's lab in Berlin. And this is a postdoc in Patzak's lab, okay, Minzer. And all of these are, so if you have interest, you can go and read them carefully. And this is the snake. It's a very pretty snake, very beautiful, and very colorful snake. and it's in Southeast Asia. It's in Malaysia, Thailand, and everywhere else, okay, in Southeast Asia. Now let's look at the problem. What's the problem? Our problem is heart failure. Heart failure is the single most common cause of acute hospital admissions of adults. People who are aged 40, they have 1 in 5 lifetime risk, and currently it affects about 64 million people in the world, okay? And it is associated with high morbidity and mortality. About 63% of people die within 5 years. And it's the leading cause of death, and heart failure costs are estimated to reach $70 billion per year in US alone, not taking Europe and other places. Okay. Of course, this is not all for the drug. I want you to understand patients with heart failure are admitted to hospitals, to ICUs. So ICU costs, the hospitalization cost, the clinician cost, surgical costs, all of these things, what all things needed will all be together close to about $70 billion per year. And the drug may be probably $10 billion or so, or less, okay? So, of course, the patients with chronic heart failure eventually end up with acute decompensated heart failure. Situation, life-threatening, abrupt dynamic. Yeah, is it better now? Okay, maybe it slipped down. Okay, so it's a life-threatening abrupt deterioration. They cannot control their heart rate, blood pressure, blood volume. That's what causes them to be admitted to ICU. You have heard of our previous finance minister, you know, who died. That's because of this problem. Okay. What's his name? I forgot. Our previous finance minister, Delhi Cricket Association's guy. Yeah. Anyway, Arun Jaitley. Arun Jaitley died because of that. And pulmonary edema, decline in renal function. So it's a very complex thing, very difficult for people to survive. You think they're just coming out, they go back again. You know, this is the problem. And course of chronic heart failure is marked by recurrent episodes of ADHF. So they just fall back and forth. From 4 or 5 times, they're gone. Okay? Rates of rehospitalization or death rate are as high as 50% within 6 months of initial ADHF event. So it's very, very quick. Okay? If you look at ADHF patients who come to the hospital, they check for evidence for congestion or evidence for cardiac output. Based on that, they are classified as warm and dry, warm and wet, cold and dry, and cold and wet patients. Okay? People who are familiar with heart failure, heart failure is also, chronic heart failure is classified as HEPREF and HEPF. So it's heart failure, preserved pressure, ejection fraction, and not preserved, low. So just to link it, warm and wet patients have increased blood pressure, low total blood volume. They have preserved or mildly reduced left ventricle ejection fraction with fluid overload. In contrast, cold and wet people have increased total blood volume but low BP. Okay, they have reduced left ventricle ejection fraction with both poor perfusion and fluid overload. So one has high volume, one has high blood pressure. Okay. So you can imagine each presentation subtypes needs specific treatment. If not, they are going to collapse. Okay. So the unmet need is for a menu of agents allowing controlled vasodilation without excess diuresis, and at one end of the spectrum, and effective diuresis without excess lowering of the blood pressure at the other end. Okay? So our molecules, modified human natriuretic peptides, potentially provide these options for the treatment of ADHF patients. So these are, there is no competing technology. These are first-in-class molecules. Okay? Let's look at what are natriuretic peptides. We all make 3 of them. Okay? ANP, BNP, and CNP. They all have a 17 amino acid residue ring, which is held together by this disulfide bond here. So this is fairly conserved. But the are quite different. And interestingly, CNP does not even have the C-terminal tail. All of these act on the heart and induce relaxation of the heart, and act on the blood vessel, induce vasodilation, and act on in the kidney. Diuresis means urine goes out. That means blood volume will reduce. And natriuresis, sodium goes out. As when you get rid of the sodium, blood pressure drops. And they inhibit renin, angiotensin, aldosterone system. So that will also control the blood pressure. So all of them, it works together. So, yeah.
Small interruption.
Yeah.
There is one pledge has to be taken by the students at this time.
Okay. Sure. Yeah. Yeah.
A small announcement. There is an order from the collector that at this time we have to take an oath regarding the human rights oath. So I request all the students to get up and raise your hands and take the oath. When I read, you can also read back slowly.
Okay. So I was telling you, it acts on the heart to relax the heart, acts on the blood vessels to induce vasodilation. That means the blood pressure is going to drop. And diuresis, it reduces the blood volume, and that's how it reduces the stress in the system. And natriuresis, sodium is gone. When you drop the sodium concentration, the blood pressure drops. And also aldosterone. That's how it does function. Okay? It doesn't go even high. Okay. So basically, by all these mechanisms, it leads to reduction in blood and cardiac load. Okay? And of course, these are all endogenous molecules, and there are also exogenous molecules found from other sources, such as snake venom. The first one was isolated from mamba venom. It's Dendroaspis snake. Dendroaspis natriuretic. It's longer tail and longer time half-life. So use this tail to create a molecule CDNP with the tail in the CP. And we isolated a molecule from crate natriuretic crate venom. That's why it's crate natriuretic peptide, and it has much longer tail, unique molecule. So we said we'll study this molecule. So just to give you how did we find it, we had a snake, not this one, but a juvenile snake, very tiny. Actually, it was less than a foot long. Very tiny snake, that's all we could get it. So from that, we isolated the venom gland and did the transcriptome, okay? This is by cDNA library, and we kept on reading more and more clones till the toxin number kind of plateaued off. So we thought that's enough. We are not interested in getting every last drop of it, but just enough so that we know what to look for. And these are all the toxins. And as you can see, it's a very small percentage, 0.44% of it is this natriuretic peptide. Okay? And of course, this sequence is shown here. So you can see the sequence is here, much longer compared to all others here, and the human ANP, which is much shorter. Okay? We got interested because there's also, one is long tail, the other one there are few changes in the ring, so which is quite interesting for us. So we said let's study. So of course my student studied this, and she did by peptide synthesis all kinds of fragments. She made one this far and one this bit, one this bit, one all this, and connected this tail to that, and all kinds of things you can do in peptide chemistry. So we made all kinds of fragments and studied everything. It's all published here. So I'm going to just tell you the ring of the crate natriuretic peptide. It binds to NPRA receptor, same receptor where the NPRA, the ANP receptor is, and uses cyclic GMP. Whereas the tail goes through unknown receptor pathway. Okay? The minute you attach the tail takes it to the unknown receptor. It does not go to NPRA at all. So tail drives the whole molecule. So that's why we called it tail wags the dog. Instead of dog wagging the tail was wagging the dog. So, and of course we were interested in only this part, okay? So today I'm going to talk to you about functional study of caring. How to study this, okay? We do in vivo studies in the whole animal, in anesthetized rats. We dissect out and expose the femoral vein and femoral artery. Through the femoral vein, we infuse drug or whatever molecules we need to do, send it. And through this femoral artery, we measure blood pressure, heart rate, pulse pressure, and everything else, okay? And in the same animal, we bladder catheterization, and we measure real-time urine flow. Is it clear? Yeah. And in the ex vivo, we take the rat aorta, cut it down into aortic rings, and put it into a perfusion bath, organ bath, and pre-contract the aortic ring, add increasing doses of ANP. It shows you vasodilation. Okay? And in an in vitro situation, we take either NPRA or NPRB, two of the receptors, separately, and transfect the CHO cells. And to these CHO cells, we add the NP analogs and measure the cyclic GMP production. So we study receptor ligand interaction, we study vasodilation, we study hemodynamic properties, and diuretic and natriuretic properties. Yeah? Today, I'm not going to tell all of this to you, but I'm going to tell you only key information, what happened in the animals. This is in the anesthetized rats. These yellow boxes shows you period of peptide infusion. We start infusing the peptide for only 10 minutes, okay, and see what happened. at low dose, this is human ANP, 0.08 nanomole per kilogram per minute. So the minute the infusion starts, you can see this is the urine flow. You can see urine flow goes up. The minute the infusion starts, urine flow goes up. And the minute you stop it, it starts going down and reaches normal. So it's almost instantaneous. The minute you infuse, it goes up and comes down. But if you look at this is mean arterial pressure or the total blood pressure in the system, it shows you at the low dose, nothing happens to the blood pressure. It remains the same. But if you increase the dose just a little bit, just times higher, from 0.08 to 0.2, it's only just 1.5 folds higher. Of course, when it's higher, it shows increased diuresis, but unfortunately, it starts inducing drop in the blood pressure. So the window is extremely narrow. Imagine you have developed a headache drug. One tablet solves the problem. You have a very severe headache, take 2 tablets, you die. Very bad, no? You have a severe headache, you take the second tablet, you die. So it's a very bad drug. So that window is very, very narrow, okay? But if you look at what happens when we infuse K-ring, K-ring is only that part of the molecule. So as I told you, we can synthesize small bits, so that small bit. When you infuse in low dose, it shows a transient drop. So there's a drop and it goes back. But if you increase the dose 5 times higher, it shows sustained drop in the blood pressure. But nothing happens with the diuresis. Almost minimal change. This got us very excited because this is a molecule, it causes only drop in the blood pressure, no effect on diuresis. So we said, let's compare the structure. So that's the K-ring, that's the amino acid sequence of the K-ring. So I assume all of you know a little bit about single letter codon, single letter code for the amino acid. And that's the human ANP, BNP, and CNP. We are looking at only this part of 17 amino acid residues. You can see these 3 residues I've shown in red are quite different, D, R, and D. They're always glycine and things. There is one arginine here, leucine here, but all the time they're glycine, okay? So we thought it's important to study what they do. So we evaluated the importance of 3 residues in the NP ring and also looked at the C-terminal tail. What we have done is we have replaced this aspartate by glycine, this arginine by glycine, this aspartate as glycine, 1 at a time, 2 at a time in permutation and combination, and all the 3 at a time, and measured everything. All the experiment I told you, we did in vivo, ex vivo, in vitro, yeah? And I'm not going to show you all the data, but from the data we could identify which residue causes what. We could identify D-link, vasodilatory and diuretic functions. We knew which amino acid substitution causes diuresis, which amino acid substitution causes vasodilation. Once we knew that, we transferred those residues to human ANP, okay? We identified this and we transferred same D, instead of glycine, we put the aspartic acid and arginine to glycine and that aspartate to glycine. So we created 2 molecules. One of them has this glycine replaced by aspartic acid, this glycine replaced by aspartic acid. It is DGD ANP, and the other one had all these 3 replaced, DRD ANP, okay? And these molecules, I didn't show you the data, but DRD ANP shows pure vasodilatory function without diuresis, whereas DGD shows diuretic function without vasodilation, okay? You'll see the data a little bit later. but this is all in anesthetized rats, okay? We also studied interaction with receptors, right? And we could see it died here. Can we replace the battery for this, please? Okay, thank you. So it interacts with only NPRA. I told you there are two receptors, NPRA and NPRB. All of them bind only to NPRA, not to NPRB, okay? But their activation kinetics are different. That means the rate at which cyclic GMP formed is very different. Diuretic analogs, ANP and DGD ANP, exhibit faster activation kinetics, whereas vascular analog, DRD ANP, it produces a cyclic GMP at much, much slower rate, almost about 80-fold, 100-fold slower, very slow, okay? So, press the button, yeah? Okay, yeah, okay. Ooh, it went up. Back, yeah, okay. Please note, all the experiments were done in anesthetized animals. In anesthetized animals, you know, anesthetics will wear out, they'll wake up. But you can't top up anesthetics because when you infuse anesthetics again, it affects the heart rate and the blood pressure. You can't keep track of it because whether it's the effect of the drug or the anesthetics. So this is not good. This initial experiment, so we wanted to study this in conscious animals. So these are animals which are awake so that there is no need for anesthetizing them. Next one, please. Yeah, that's one, yeah. So we studied the effect in conscious normal sheep and heart failure model of sheep. And this work was all done in Christchurch Heart Institute. As I told you, Miriam did the work, and Mark Richards was the director of CVI, Cardiovascular Research Institute in Singapore. Also, he was the director of Christchurch Heart Institute, okay? So, and in these animals, you could study full hemodynamic profiling, all the blood pressure, heart rate, everything else, and full renal assessment, how much urine goes out, what kind of ions go out, what happens in the urine, everything we could measure, and full species neurohormonal immunoassays. What are all the changes that happen? Because we want to understand how does it affect the animal, okay? So all of them, data is all published. So it's in cardiovascular research paper journal. But I'm going to show you what happens in the normal sheep and heart failure sheep only with the blood pressure development. I wanted to follow the thing. D1 is the low dose, medium dose, and high dose. All these doses of peptides were infused for 90 minutes each. Very long time. And the doses are given there. So if you look at the black circles, as you increase the dose of A and P, you can see the blood pressure drops. The minute you stop it, it goes back. Similarly, the red triangles show you the DRD, A and P. As you increase the dose, the blood pressure drops, and the minute you stop it, it goes back. The blue colored squares, they're all DGD, A and P. It almost follows the cell line, which is control vehicle, which is in the open circles. That is true in normal sheep as well as in heart failure sheep. So more or less nothing happens. With the DGD, it does not induce any drop in blood pressure. Although the dose is very high, it's about 10 times higher compared to DRD, ANP, and ANP. So it's much higher concentration, but it does not drop the blood pressure. If you look at the urine output, diuretic property in the top row, and in the amount of sodium going out in the urine in the bottom row, and normal sheep and heart failure sheep, I want you to focus on the black histogram and the blue histogram. Black is ANP, DGD ANP is the blue one. The red color one is the DRD. Nothing happens with the DRD, whereas with the black and blue, you see as you increase the dose, it goes up. Yeah? So it shows increased diuresis and natriuresis. So DRD ANP produce vasodilatory effects very similar to those of ANP in both normal and heart failure sheep models. In contrast, DGD ANP had negligible effect on arterial pressure even at 10-fold higher concentrations. And DRD ANP produced no significant renal effects in either normal or heart failure sheep. In contrast, DGD ANP produced comparable increment in urine volume and natriuresis, sodium excretion, similar to ANP in heart failure sheep. So you could see a clear separation of function between the two. Okay? Next, please. DRD ANP has entirely renal neutral profile. So it does not do anything in the kidney. So it's warm and wet, suitable for warm and wet ADHF cases. This is the most common. About 49% of ADHF patients belong to this group. And 30% of patients with warm and wet cases, they die within 18 months. These patients have established or threatened pulmonary edema with preserved blood pressure. They have blood pressure is high, but blood volume is not. Okay? DRD ANP redistributes the circulating volume from the congested place. In a pulmonary edema, it pulls them all out and relieving the pulmonary edema with minimal risk of excess volume depletion because it does not cause diuresis. Okay? Next, please. So DGD ANP retains full kidney function, and cold and wet cases is where you can use it. This is only 20%, but this is the most lethal part of the cohort because 50% of people die within 18 months. And these patients have increased total blood volume with edema in multiple compartments. They are hypertensive. Their blood pressure is low. You don't want them to drop the blood pressure. That will collapse them. Okay? So when the blood pressure goes down further, the blood don't reach every part of the body. So that's a problem. So DGD ANP resolves edema in lungs and periphery without lowering the arterial pressure and with attendance risk of hyperperfusion and renal failure and shock. For the first time, specific pharmacotherapies are developed to manage ADHF and better management to reduce time in hospital and in ICU, more confidence in dosing and reduced risk of hyperperfusion and underdosing. So they're good to do treatment for heart failure patients. Now the question is, why such a subtle small change makes a difference? So we wanted to understand that. So we worked with Professor Ogawa in Kyoto University. I wanted to understand, I kind of told you that networking is very important. In our lab, in NUS, we can do cryo-EM studies. We can. But we wanted to work with Professor Ogawa because they've already created the clone and studied X-ray crystal structure. And we thought it's very easy to get their help and interact with them. When you interact with them, of course, it's 2 + 2 is not 4. It will become 5, 7, 8, okay, because there is also new ideas coming, new strategies coming. So we went and studied with them. So they did, you can see the extracellular domain of this molecule, which is bound to human ANP, DGD ANP, and DRD ANP. So these are the cryo-EM structures of it, solved at 2.6 to 2.446. And when you pull out this and look at the density, you can see the density of all these three. And next, please. He is getting tired. So you can see there are subtle changes. I'm not going to describe you all the subtle changes in that, but it tells you how insertion of residue, 2 residue, pushes something and pulls something towards it. So that's how the whole molecule changes. Next, please. minor sequence changes drastically affects the conformation of the analogs. Okay? Next, please. So if you look at the structure, I'm not going to explain all the nitty-gritties of it. It shows a lot of differences, and NP analogs induce distinct conformational changes. If you remember, I kind of told you ANP and DGD ANP produce cyclic GMP much faster, whereas DRD is slower. So we know what happens in the extracellular domain, but cyclase domain is in the cytoplasm. We don't know how that information translates into that. Of course, we don't have the full length, so we went to some theoretical ways of doing it. So this is the full length molecule. They actually structural changes on extracellular, that's real. What happened in our molecules, we know that. But bottom one is pretend. Pretend is hypothesis. We don't know, because there is adenyl cyclase structure known, and we have used that. Thank you. Sorry to trouble you. Yeah. Okay. So all this, what all changes you see here is actual structural changes, and what you see here is pretend. We don't know exactly. Okay. It's just a hypothetical changes based on actual structure which is determined in adenyl cyclase, not guanylyl cyclase. Okay. There is subtle difference between the two. Okay. So when you do human ANP, it does bind together and cause this activation of this adenyl cyclase. GTP is converted to cyclic GMP, and when DGD binds into this, it just drags it a little bit that side. Okay. Because of that, it slows down the cyclic GMP production. And when you add DRD ANP, it actually pushes this molecule quite a bit this side, leading to slower cyclase, formation of cyclic GMP. Of course, this is just a pretend. We don't know. We want to do structures of ANP and its analogs with full-length NPRA to understand exactly what happens. Okay? This is only pretend cartoon. Okay? Don't trust those things. So we designed two molecules for warm and wet and cold and wet separately. The human ANP, I want you to understand, it induces diuresis and drop in the blood pressure. But DGD ANP, only diuresis, no vasodilation. There are only 2 amino acid changes. And with 3 amino acid changes, DRD ANP induces only vasodilation, no diuresis. Difference between the two is only one glycine replaced by arginine, one residue. One amino acid residue leads to 2 opposite ends of spectrum of pharmacology. Very different. Okay? And altered kinetics of cyclic GMP production and structural changes in the ligand, and it induces distinct conformational changes in the receptor extracellular domain at this point. Okay? Basically, substitution of key residues determine subtle interaction with NPRA, leading to distinct pharmacological subtypes. So let's look at what happens in the kidney. Okay? I know do I have time? Yeah, okay, thank you. So in kidney, as you know, glomeruli, plural, so they are the ones where filtration occurs. Okay? So if you take it out, this is afferent arteriole, which brings the blood into the glomerulus, and that's the efferent arteriole, that's where the blood leaves. Okay? These can be studied in anesthetized rats. It's called hydroaortic kidney models. They slice the kidney in the anesthetized rats, open it, clean up the parenchyma, and you can measure, you can look under the microscopy, you can start seeing how the blood vessels enlarge and things like that. And in the animals, what they have done is they have infused ANP. When they do that, this afferent arteriole becomes dilated. It increases in diameter. That means more blood can come in. And this one, efferent arteriole, induces vasoconstriction. It becomes very narrow. So more blood comes in, less blood leaves. What happens? Blood pressure increases here, right? Because more blood can come, less blood can leave. So there's a pressure. So that causes urine to filter out. So that's the glomerular filtration rate. And more urine is formed. Of course, this is in a whole animal. In the whole animal, you can see there are so many feedback mechanisms. We don't know whether it's because of the feedback mechanisms or ANP is doing it by itself. So we wanted to understand. I'm a biochemistry person. We want everything to be dissected out into small things to find out exactly what's going on. Yeah? So we isolated this. This is mouse, glomerulus. And that's the afferent arteriole, that's the efferent arteriole. So click please, yeah. So, okay, it's okay, it's okay. It's okay. It's okay. Don't worry, don't worry. No big deal. Nothing, no harm. Yeah, okay. Can you click that? Yeah. So this is afferent arteriole. What we are doing is we add the angiotensin II. It induces vasoconstriction. And on this vasoconstricted molecule, we add increasing amount of ANP. You can see as the ANP increases, it becomes dilated. It becomes larger and larger. And on the efferent arteriole, we don't add the angiotensin II. We add increasing amount of ANP. As you add, click please, yeah. Oops. Yeah, go to inside the figure, Yeah. When you go here, yeah, now you can see this, right? Click the arrow, please. Yeah, yeah. As you increase the ANP, you see it becomes more and more constricted. Okay? Yeah? I want you to understand there's still flow going through inside. So it cannot completely close. Okay, there's still flow. Okay. Good, yeah. Click. Yeah. Yeah, one more, one more. Yeah. So this is what already you saw. Unfortunately, video didn't work when we connected. So I had to open it and show you. And go to the next slide, please. Thank you. Yeah. So what we have done is, if you imagine that's the original luminal diameter, inside diameter, and the minute you add angiotensin II, it induces vasoconstriction, and as you add increasing amount of ANP, you see it gets dilated more and more. It almost becomes as close to its normal diameter. Okay? But nothing happens in the control, the red one. And nothing happens in the efferent arteriole. Not much significant happens. But if you look at the vessels without preconstriction, we don't add the angiotensin II. But in the efferent arteriole, as you add increasing amounts of ANP, you see vasoconstriction. Nothing happens in the control or the afferent arteriole. It tells you that ANP induces vasodilation in afferent arteriole, vasoconstriction in efferent arteriole. What they observed in the animal is correct. It's nothing to do with any feedback mechanism. It's everything. One ligand do that job. One side it induces vasodilation, other side it induces vasoconstriction. So if we had all these analogs in the preconstricted vessels, that's the ANP, and DGD ANP also induces vasodilation, not exactly full, but quite a bit, but DRD ANP, no effect at all, almost nothing, yeah? Whereas nothing happens in the efferent arteriole. This is the afferent arteriole, this is the efferent arteriole. So only ANP and DGD ANP induce vasodilation in afferent arteriole. ANP is better than DGD ANP. They are way better than DRD ANP. But if you look at the vessels without preconstriction, nothing happens in the afferent arteriole. In the efferent arteriole, as you increase the dose of ANP, you see the vasoconstriction, and DGD ANP slightly lower, but it does, and DRD ANP also does a little bit, but even lower than the DRD ANP, DGD ANP. All three peptides induce vasoconstriction. ANP is better than DGD, ANP better than DRD, ANP. But it tells you function is determined by rate of cyclic GMP production, because faster the cyclic GMP, more is the constriction. Yeah? But fine tuning of cyclic GMP is important for increasing the GFR, right? So in a cartoon form, that's the glomerulus, afferent arteriole. When you add ANP, it induces vasodilation, induces vasoconstriction here. So because of that, more blood coming in, less blood leaving, you have an increased GFR and increased diuresis. That's the same thing as DGD ANP, slightly lower compared to ANP. But DRD ANP does not induce vasodilation. It does very little vasoconstriction. So it has minimal effect on GFR and diuresis. So observed functional difference explains distinct diuretic profiles of the animal. In the animal, what you observe. Question is, why receptor ligand pair show opposing effects in afferent and efferent arteriole? You know? And this is the first time, first example in nature, one receptor and one ligand, just because it's acting in different tissues, it shows completely opposing effects. Okay? So when we wanted to study, when the ligand binds, we said it forms cyclic GMP. We wanted to study the effect of cyclic GMP on efferent and efferent arterioles. So this is precursor. As you increase the doses of cyclic GMP, it shows vasodilation. Nothing happens in the efferent arteriole. In the vascular preconstriction, increasing cyclic GMP shows vasoconstriction, but nothing happens in the afferent arteriole. Basically, similar to ANP, cyclic GMP induces vasodilation in afferent arteriole, vasoconstriction in efferent arteriole. It tells you signal transduction pathways downstream of cyclic GMP are different in afferent and efferent arteriole. Same cyclic GMP produces vasodilation in one vessel, vasoconstriction in the other. Okay? So distinct subtypes of smooth muscles are there in afferent arteriole and efferent arteriole. One of them is phasic in afferent arteriole and tonic in efferent arteriole. Currently, we don't know the molecular differences between these two types of muscle fibers. Okay? When you compare ANP versus cyclic GMP, ANP almost completely full dilation, but this one you can see only 75 to 80%. You see it's plateauing. And at very high dose, it's 100 micromolar concentration. It never reaches that high. So that means it's not reach the final full dilation. It could be because cyclic GMP enters through diffusion. It has to go through slowly. And accessibility could be one problem. And of course, phosphodiesterase reduce the concentration of cyclic GMP. That's why it doesn't reach. Whereas ANP induces cyclic GMP production, which is released in situ. And ANP and DGD ANP, they produce cyclic GMP much faster, and they can induce dilation because they reach a threshold. Okay? Whereas DRD ANP, it produces cyclic GMP slowly. As and when cyclic GMP is produced, it's getting chopped up by phosphodiesterase. And because phosphodiesterase clears the cyclic GMP, there is no dilation at all. It doesn't reach the threshold needed for the dilation. That's our imagination though. So vasodilation depends on the kinetics of cyclic GMP production. ANP and DGD ANP can show that, but DRD ANP does not show. Okay? So in cartoon form, you can see how vasoconstriction, preconstriction when you add angiotensin II, and when you add ANP, cyclic GMP is produced, induces relaxation of the smooth muscle. That's the same thing with DGD ANP, but this is slightly lower amount, whereas DRD ANP produces even much lower amount of it. As it comes out, it's getting chopped up, and therefore it never reaches the threshold to do that. But when you add cyclic GMP, it will come in, but it may not be sufficient to reach the same concentration as you want in situ. So if you compare in the efferent arteriole, it induces vasoconstriction ANP and the cyclic GMP, more or less to the same extent. You will never be able to go to 100% because you are looking at the luminal volume, so you cannot go to 0, okay? And PDEs do not seem to affect cyclic GMP concentration in efferent and afferent arteriole. Constriction depends on kinetics of cyclic GMP production. So this is how we explain all this. So basically we did show that cyclic GMP is responsible for all this, and the story continues. We are still interested in many parts of it. I'm going to skip a little bit because I've taken too much time for whatever reasons. I'm going to tell you ECD confirmation is important. This is the future of what we want to do, and how that affects the guanylyl cyclase we want to understand. Of course, we also want to understand how cyclic GMP induces vasoconstriction, and there is hardwiring, which I didn't tell you the part of the story, but role of protein kinase G, PDE, and cyclic GMP gated ion channels we want to study. There's qualitative and quantitative proteomics and phosphoproteomics we would like to study. Of course, when you do this, you start getting more questions, okay? So like that, this saga continues for you to continuously understand something new, something different, okay? With this, I'm going to stop here. I would like to thank you for your patient listening. Thank you. If you have questions, I'll be happy to answer. Questions? Yeah. Yeah, go ahead. I'll tell them. Question is, we have tested it in mouse glomerulus. Is that what happens in mouse, man and sheep? So most likely, yes, it works, but we have not tested them correct because it is very hard to get human kidney to do it. Sheep kidney, we didn't want to do it because it's the same kind of thing. They are very related. You observe the same thing what you observed in all the three sets of things. So we hope to see the same thing, correct.
ANP is functioning only in kidney and heart and not in any other places?
There are, but this is, see, when we measure the peripheral blood pressure, you are not actually measuring the aorta dilation. Actually, dilation has to happen in the arterial beds of mesenteric things, and there are many other arterial beds where arterioles have to expand. But it's very difficult to identify which one is the right kind of arterial beds to do it. So I've talked to many people. So it's a very complex one, but we don't know how to go and study them at this point.
Why this question came to me is, is because in the reproductive system, the vasodilation in male is having a major role in fertility or...
Yeah, correct.
So that's the reason I asked.
Correct. No, it's a very important question. He is trying to relate vasodilation to penile erection and all. There are so many other implications in many places. And also there is antidiuretic hormone. We don't know how this reacts with the whole system. So there are still a lot of things, but in the whole animal, as we have shown, one induces pure diuresis, other induces pure drop in the blood pressure. I mean, as pure as possible. I mean, it's very difficult to have 100% accuracy, right? Yeah, correct. Questions? Students should ask questions. Any question is fine, don't worry. It's a tough crowd. Yeah, please.
Sir, I have a simple question.
Sure.
So we are searching for medicine in toxin proteins. I could not understand the connection between finding the medicine from the toxin proteins.
Yeah.
So why we should go for finding medicine? Why should go select the toxin proteins instead of other proteins?
Yeah, you can go for other proteins. So one advantage of toxins is designed, evolved through millions of years to target the prey, to interfere in a physiological system. And it's all concentrated in 30, 40, 80, 100 proteins. But if you imagine I squash up liver, kidney, neuron, muscle, whatever, each cell has about 6,000 to 10,000 proteins. Almost all those proteins are important to make neuron a neuron, liver cell a liver cell, kidney cell a kidney cell, muscle cell a muscle cell. If I take a protein from muscle cell and drop it onto neurons, nothing happens. It's not important at all. Okay? Similarly, neuron protein, I drop it onto liver or muscle, nothing happens. However, there are some molecules, as you have seen, hormones, and there are some secreted ones, cytokines, chemokines, and things. They would go and attack and do something somewhere. These are very small percentage. Imagine these are in immune cells, there are probably 10, 12 molecules. All the interleukins, chemokines, and things are released. Not at the same time, different times and things. 10, 12, 20 of them, for example, out of 10,000 will be useful. So from 100, 10,000, you're going to look for 20 is a very small number. Whereas all the toxins have evolved to do a job. If they're not there, the animal is going to die.
So now I could see the connection between
Correct.
your logic, sir.
Correct.
The toxin proteins and the medicine.
That's the reason.
Thank you, sir.
Thank you. Yeah, target specific, function specific. So that's why I kind of started saying why we target animal venoms, why we target blood feeding animals. Of course, that doesn't mean there are other things which are not there, but we have a limited time, so yeah, correct. Yeah. Questions? No students have any question? Did you all understand what I said? Yes? To an extent, not fully, but 80, 90% of it. So you should have 10% questions, right? What is that? Why did we do what we did or something? No? Nothing bothers you? Don't hesitate. There are no stupid questions. There can be stupid answers. If there are no questions, we stop here. Thank you. Thank you very much.
I would like to extend my sincere gratitude for your fascinating and informative presentation on Vinome today, sir. Your talk not only deepened our understanding of the intricate mechanism of Vinome, but also highlighted the exciting potential for Vinome-derived compounds in medicine. And I'm sure that everyone in attendance left with a newfound appreciation for the complex and fascinating world of Vinome. Thank you, sir. Please enjoy a short tea break before the next segment and assemble here for the next session on 12:15 a.m.
esteemed resource person. The floor is yours, sir.
The resource person for the second session is Professor Devarajan Karunagaran, Emeritus Professor, Department of Biotechnology, IIT Madras. He completed his undergraduate degree in chemistry at the University of Madras and Masters in Biochemistry from Jibmar, Pondicherry. He acquired his MPhil and PhD degree in Biochemistry from Krishnadevaraya University, Anandpur, Andhra Pradesh. He joined in IIT Madras in 2006 and pioneered cancer research there. He played vital role in laying the foundation for a nationally recognized cancer biology program in the institute. At present, he training 16 doctoral researchers and guiding 15 PhDs and 5 MS students. His contribution in understanding the cancer biology have been recognized by the various prestigious awards. He is a recipient of C.R. Krishnamurthy Lecture Award and Kinariwala Research Award from Gujarat Cancer Research Institute, Gujarat. With this, I invite him to deliver lecture on a novel therapeutics approach in breast cancer.
Thank you. Very good afternoon to all of you. I take this opportunity to thank the organizers, especially Victor and others who invited me to come here and give a talk. As you might have noticed, I started my career in this college, this auditorium, and it was built, I was here working as a lecturer in the Department of Biochemistry. So it is natural for me to have an attachment with this place. It is like coming back home. After all, I was teaching here for more than a decade. 13 years I was teaching here, from 1979 to 1992. Anyway, with this brief introduction, let me go on with this topic. Although we have published research papers and all that, for the sake of a student, I will be telling certain basic things in the first part of the lecture, so that you will be able to appreciate this area, because what I am going to talk about microRNAs, maybe people are not very familiar with that. So let me begin with some simple things. How do I move the slides? Isn't it? Yeah, no. When I come here only, it moves. Yeah, just. No, that's okay. Only when it is near, it works. It's okay. I will manage. Don't worry. I will manage. Yeah. All of you know that let me come this side. It's easier for you. Human genome. What is human genome? The total genetic content in a human cell. In a cell, if it is a rat cell, you are going to call it as a rat genome. If it is a mouse cell, you are going to call it as mouse genome. So the total genetic information. So people say that about 25 to 30,000 genes are there. But interestingly, in biology, I hope you know about this. I will repeat that anyway. All these cells that we have, kidney, stomach, whatever tissues you can imagine, all of them have these genes, but then they are totally different. Why is that? They have the same genes. The genetic content is same in all the tissues, but they look different, they function different. Why? Because these genes are expressed differently in each tissue. So a liver tissue is a liver tissue because it expresses a certain set of genes. A kidney tissue is a kidney tissue because it expresses certain other set of genes. Although all of them have this potential, this has to be understood for what I am going to talk. That's why I am just telling this. So, by changing the gene expression, theoretically speaking, you can change the tissue also. All of us, for example, are born by the fusion of sperm and ovum, and you call it a zygote. When you look at that zygote or the initial stages of the embryo, it looks an amorphous tissue. You don't make out anything. But after a few months, in the case of humans, you start seeing eyes, nose, all these things. You call that as differentiation, right? But for this differentiation, is there any mutation? It is the same genetic content, but then it differentiates into different tissues. Why? Because of changes in gene expression, which immediately means to you that if you change the gene expression, then you can change the tissue itself. So changing, or anybody who touches any factor that touches gene expression can do a lot of things. So this is the basis for our talk today. Why, I will tell you. In the human genome again, all of us, when we say about genes, what are genes? Normally the teachers tell you that the genes are those which encode proteins. What is a gene product? Protein. That is what people will say. But if you look at the definition of the gene as we have evolved, it is not that it has to always produce a protein, right? Genes may encode something, a functional product. For example, if something is a gene, it may produce an RNA. Maybe it doesn't produce a protein. Even then it's a gene, right? So the definition keeps changing as and when you acquire knowledge. But if you see the human genome, as I told you, 25 to 30,000 genes are there. In the total human genome, how much is actually translating into protein? It's only 1.1%. So what happens to the remaining? We have a tendency to say anything we don't understand, we will say junk. Even in the class, as I was teaching in this very same college, when I was teaching actually, we had 13 faculty members in the department, and there was one student, I don't want to name, that boy was a little bit, in those days it was very different. Probably today things have, you have got relaxation, but this boy was smoking, this, that, and so people had a very different opinion. All the faculty members were almost saying that this fellow was useless. Okay. But then this boy came to me, he did MSc here, and came to me and asked, sir, I really want to do a project. I also had the same opinion that this boy is a bit deviant, he is just not a very studious boy, so he may not do things sincerely and so on. But then his kind of perseverance, that he came again and again and asked me about it, I took pity on him and agreed to give him a project. Probably Mohandas knows, by around that time. Mohandas also was one of my project students. It's a very similar project only I gave. So I gave him that project, and I had this doubt whether this boy will really work. I was living close by here, somewhere near the Goldwinds. So I took the scooter one day. You won't believe, I came here to the college by 2 o'clock in the morning and saw whether whether the people are working in the lab. And he was working. So I got impressed. Okay, this boy is not, after all, not so bad. And my impression was correct. After he completed here, although he failed a few subjects and made up, all those things are there, and he was not very good also. But he still had an interest, and he went to Australia and became a good molecular biologist. What I am trying to say is, teachers have this tendency to brand somebody as bad or something. It's a very wrong thing. Whoever does that is a wrong thing. Each one has a potential. So also this DNA. People immediately said that we don't know the function of this DNA and branded that as junk DNA initially. Even people you can see in the textbooks, they will say dark genomic matter. Dark means you don't know anything because it's dark. You are dark about it. It doesn't mean that is dark. You are dark about it. You don't know about it, but it still has some functions. So everyone, my feeling is everyone has a potential. Similarly, I will tell you a definition of a teacher. I always said this. Teacher itself, to me, it's a misnomer because it doesn't define what he does, he or she does. Because basically for me, teacher means a learner. So he is also a learner. If I have come here, you may not know, I may have learnt something from you, which you may not believe. But I can tell you stories about that also. After completing 13 years in this PhD college, I went to Israel. At that time, I completed PhD, all right. I went as a postdoc, but I was not fully trained on modern things. Even modern computer, it was not known to me. I just knew there is some computer, but I couldn't use the computer even at that time when I went to Israel in 1992. And I was trying to do Western blotting. All of you know that Western blotting is an equipment that is used to study the expression of proteins, right? Or let us say. So that technique, I didn't know. And when I asked the students there, PhD students and postdocs, they were not willing to teach. My boss told me, here is a student who came here to do summer training. She was a school girl. At that time, she was around 16 or something. I was already more than 40. So she was like my daughter. So my boss told me, you learn from her. I was very much happy. I learned Western blotting from that girl only. She was hardly 16. So I learned a little bit of Hebrew and used to tease her. Morash Ali means my teacher. So every day I will say, Bakarto, good morning, and my teacher is coming. And she was very happy. So a friendly relation made me to learn these techniques. What I am trying to say is, you should be a teacher, real teacher should be able to learn from anybody and everybody and will keep learning as long as he or she lives. So that will probably a definition of a teacher. Okay, I will keep on telling like this, but the idea is that nothing is a junk and everyone has a potential like in humans also. So these also had some potential. As you can see, the rest of the DNA transcribed into RNA 25%, whereas what is being translated into protein is only 1%. Remaining 99% is not a junk. People started identifying functions. For example, post-transcriptional silencing of active genes. Now I am coming to the point. So in that 99, what was thought to be a junk, you have non-coding RNAs. They can silence genes, and just now I told you, if you can silence the genes or if you can regulate the gene expression, you can do so many things. I am a cancer biologist. In cancer, we say that the normal cells, after all, cancer cells are not alien cells. They are your own cells. Something happened to them and then they got transformed into cancer cells. So your own genes got deranged into cancer genes. So the idea is, many people believe that many mutations have happened and that resulted in the transformation of normal cells into cancer cells. That's a mutation theory, we say. But then one can always argue, just like I told you during development, you don't need mutations to change the normal cell into a cancer cell. One can argue like that. People are arguing like that also. That is called a tissue regeneration theory, something like that on cancer development. What they are trying to say is, the importance is, if you touch the gene expression, you can do, you can change the tissue itself, tissue identity itself. Another example I can give, stem cells for example. You take fibroblasts or any other cells, if they are stem cells, you can convert them into brain cells. Are you mutating them? No, you are not mutating them. All you are doing is, you are changing the expression. So changing the expression and touching the expression is very important. That is why these are tiny players, the so-called non-coding RNAs. Although they are small in structure, they do a big job. So that is why you cannot ridicule a person or anything because of the size. You have to see how it works, what functions it carries out. Okay, without saying so many blah blah things, these people they found out already that the post-translational gene silencing siRNA, small interfering RNAs, you must have heard about. These discoveries earned Nobel Prize already in 2006 to Andrew Fire and Craig Mello. Okay. Actually, this theory of RNAs suppressing the genes, this theory was there somewhere around 1990. Rich Jorgensen at the University of Arizona, they were trying to do some experiments in plants. What they were doing is called petunia flower. They were trying to see this color, some you can say violet or purple color. This is somewhat dull when you come to this. So they wanted to deepen this color. So what they did is they introduced the purple gene into that and tried to see whether they will get a very deep purple color flower. But to their surprise, they never got something like this, they got something intermediate also. They didn't get this or this, somewhere intermediate they got. So they called it as co-suppression. I am not explaining purposely. This phenomenon was called co-suppression, since both the expression of the existing gene, initial purple color, and the introduced gene, the deep color, were suppressed. So they were getting somewhere in between. So that was called co-suppression. Later on, some other people, they did some experiments. It is, that is a starting point. What was done in plants was the starting point. Then they understood that RNA can do something to suppress the gene expression. So that was the idea. That is how siRNAs were born. I am not giving the details, but here they have introduced sense RNA and then antisense RNA. They did not see the expression of the gene disappearing. But when they introduced double-stranded RNA, they saw that the expression of the gene disappeared. That means they were able to silence the gene expression with not a single-strand RNA or sense or antisense RNA, but with a double-stranded RNA. So this is how siRNAs were discovered, and they got Nobel Prize also for that. Now, these experiments were demonstrated in C. elegans, which is a worm. Once you don't have the, when you successfully knocked out a gene, in this case a particular gene, it will result in twitching. So that was a measurement. So you have to have a phenotype to see whether the gene was suppressed. So when the gene is suppressed, they know the C. elegans will start twitching. So when they did these experiments, they saw this twitching. So twitching was the end point for them. So this is how they have discovered that. Now this year, 2024, two people got Nobel Prize for discovering a similar phenomenon of non-coding RNAs, which are what is called microRNAs. They are 21, say 19 to 25 nucleotide in length, small RNAs, which are able to suppress the gene expression. Now I am coming to the point. That is this Victor Ambros and Gary Lukin. This is again an interesting thing there. In the 1993, I think, I have a slide over that. Yes, somewhere in 1993, Ambrose and colleagues, they found what is called LIN4 unexpectedly did not encode a regulatory protein. Gene means that you usually think that it will encode a protein, but this LIN4 gene was not encoding a protein. So they were a bit surprised at that time. Instead, this LIN4 was able to suppress the expression of, especially its messenger RNA, was able to suppress the Lin14 protein. So they checked certain sequences. They found that the Lin14 messenger RNA, I hope you know what is 3' UTR, untranslated region, had some complementary sequence to Lin4. So they hypothesized that probably by complementarity, this Lin4 bound to the 3' UTR of Lin14 and suppressed the expression of the mRNA. Once the mRNA is not expressed, protein also will not be expressed. So this is how they discovered. They called it as microRNAs. Nobody bothered. Although I told you the siRNA story, all those things, nobody bothered that this is down-regulating this expression of Lin14 and all that. That is mainly because these were discovered in C. elegans. We have a tendency, again, our own tendencies are very funny. We think that medicinal discoveries will come only, the people will say, some doctors, not to offend them, but they are right also in one sense. What you do in mice is not going to be applicable to humans, they will say, because mice is not human. They are right in that. It's not that they are wrong. But everything you do, you can learn some lesson and can proceed further. That is what the science has shown us. Here they have done it in C. elegans, nobody bothered. Now in 2000, another C. elegans microRNA LED7 was discovered. This time they found that this is conserved, these kind of genes are conserved across the species. Then everybody jumped up. So 7 years people waited. First microRNA was discovered in 93 itself, but up to 2000 people waited because they find an evolutionary thing. Somebody said, nothing in biology makes sense without relating it to evolution, they said. So once you make it, why? Because once you relate it to all the species, then there will be some use by studying this. You may be able to relate to humans or agriculture or some other useful things to humanity. That is why people are very much interested in this. So in 2000 only another LED7 microRNA was discovered, and then this started going up. So people realized that these small RNAs, microRNAs, are highly valuable molecules because they alter the gene expression. Usually they are silencers of gene expression. Remember, just now I told you a few minutes back, if you touch gene expression, you are almost playing God, because you are able to make tissues, tissue identities. You are able to play with the system, organism. So that is very, very important. That's very important to understand. So, immediately people realize that it could be used in development, physiology, it may be in disease and cancer. These tiny microRNAs could be useful. Now we will come to that. What are these microRNAs? These are non-coding RNAs. Just like RNA, all of you know messenger RNA, transfer RNA, ribosomal RNA, the microRNA. People have discovered piRNA, so many other RNAs people have discovered. But these RNAs are like, they have their own genes. There are about 2,500 microRNAs in the human genome. All of them, how an RNA is produced? From DNA. That is what you call it as transcription, right? From RNA, if you can make protein, you call it translation, right? So they have their own genes. About 60% of them have their own genes, microRNA genes are there. As I told you, about 2,500 microRNAs are there in the human system. So when they are produced, actually they are produced as bigger molecules, what are known as pre-microRNAs, at a bigger length, 70 or even more. Primary microRNAs first, and then pre-microRNAs, and then only mature microRNAs. So these are all taking place in the nucleus. And they have different systems. They they are produced with these tails. You can see this primary microRNA. See, this is a single stranded RNA only, but it becomes, it looks like double stranded because it folds itself into a stem like structure. So it looks like a double stranded RNA because it is folding, and it also has a tail. So all these are processed by various enzymes, microprocessing. They are cut and then they are transported across the nucleus, and then they come into the cytoplasm where they encounter what is called a RISC complex, RNA induced silencing complex. There you have certain proteins, enzymes and all that, and ultimately they come out and they can bind with, they first come out as double stranded and then each strand separates. One operates as a microRNA, the other one acts as a passenger strand and so on. So people call it differently, but latest research shows that even both the strands can function as microRNAs. That is one of the latest developments. Let us not confuse that. So a single stranded RNA only ultimately acts as a mature microRNA, and that binds with a sequence that is found in the 3' untranslated region of the mRNA. Now you may be wondering, sir, you are telling something about siRNA, you are now telling microRNA, what is the difference? siRNA is something different. It is only against one gene, right? You are silencing one gene. Here, interestingly, depending upon the sequence in the mRNAs, it can practically target about 500 mRNAs. Don't get confused with mRNAs and miRNAs. miRNAs target mRNAs, and there can be 500. Actually, very interestingly, after I left this place, when I went abroad and got trained and all that, I went to Trivandrum to work as a scientist. The scientific institutions, the CSIR institutions and all that, there is one problem there. You have to work on a particular problem, you cannot deviate. There is some dictatorship there. There is no academic freedom in one sense. Okay. So if you are branded as a cancer biologist, you have to work only on cancer. But then I moved to IIT. IIScs are thought to be very good in the sense academic freedom is there. So the moment I went there, I thought I should do something new. You remember, I went there in 2006. At that time, if you remember the discovery of microRNAs, I told you it was only discovered in 93, but then in 2000 only people started waking up to the realities. And so 2006 was not far away. In science, 10 years is not big. So 2006, these things were coming up. It was a frontier research. So I jumped into that without knowing anything, which is good. You don't know in science sometimes when you are an expert and want to do something, you are going with a preconceived notion and you may make mistakes because you cannot do experiments because you are assuming the result. You cannot do research like that. You should not do actually. So when you are going with an open mind, since I was a zero in this, I was able to do something. So I went there and started this area, microRNAs, to do some research in IIT. And IIT gives freedom. You can do whatever you want. Nobody prevents you from doing anything. So we started on this. At that time, the point I am trying to say is, we were biologists. I was trained in biochemistry. Most of us do not know maths and other these things, computational things and all that. Whereas IIT students are good in that. So I thought we have to make use of them and called some students. They came for projects and I told them, since these are complementary, the microRNA binds to the 3' UTR of the messenger RNA. So this is all based on the sequence. So you can predict the targets of the microRNA. Similarly, if you know the targets, you can predict what microRNA will act on that. So I told the student, at that time we didn't know much. So I told the student, Next day, he took one microRNA, just randomly picked, and came out with some 300, 400 targets. So I was confused. I didn't know what to do, how to work on that. Since my previous track record was on signaling, NF-kappa B signaling and all that, I told him to look for some signaling molecules and look for microRNAs that will target them. That is what I told him. This is how I started to do research on microRNA with zero knowledge on that. And he came out with some things. So then I became very excited. Okay, now we have bioinformatic tools. At that time, tools were there. Many softwares are there because this is a simple prediction based on this Watson-Crick base pairing. So once you know the microRNA sequence, it's very simple sequence because 18 to 25 nucleotides. You just see the complementary sequence in the genes. Again, gene database is there. 25 to 30,000, all these mRNAs are there. Their sequences are there. UTR databases are there. So you simply look at it, and there are also softwares available. All you have to do is put the sequence of the microRNA, you will get the targets. Put the target sequence, you will get the microRNA. So this was as simple as that. So we were able to get some, and also they were giving some scoring depending upon the binding constants and the dissociation constants and the temperatures, oligonucleotides, the temperature, annealing temperature. Based on that, they have given some scoring and they will say this is the best target. Bioinformatics people, the software. So we got the best targets and best genes. So since I worked in Israel and other places, I had friends, so I called them and asked them to give some microRNA genes to express in the cells, cancer cells. And we were able to do that very nicely and find out the targets. Nothing worked out. For nearly 2 years we were struggling and we were not able to get the targets. What happened is I realized what you do in vitro or bioinformatics may or may not work in vivo. That is the lesson I learned. Then I came out with some other ideas and finally succeeded. I will not have time to tell all those things. Anyway, so they come out of the nucleus, bind here, and then they will inhibit the expression. Similarly, they have an interesting thing. They will also enter into multivesicular bodies like exosomes and can be secreted out into the bloodstream. So microRNAs are capable of, you can get them in the blood system also ultimately, exosomes also. That's what I want you to understand. Now, I will come to the next question. Sorry, what's happening? It's not moving here. Moved up. One more. Yeah. Just like other genes, these genes that are encoding microRNA, they can also get abnormalities. They can be epigenetic alterations, they can be deleted, they can be post-transcriptional modifications and so on. So if there are defects in the genes that are produced in, that produce the miRNA, you can have defects in the miRNA production also. I will tell you one important thing. Sir, you are a cancer biologist, why are you talking about microRNAs? The reason why cancer biologists work on microRNAs is very simple. In cancer biology, there are two important genes, oncogenes, tumor suppressor genes. All of you know. Oncogenes are growth promoting genes, tumor suppressor genes are, they are going to suppress the cancer genes, right? Now, imagine microRNAs are gene silencers, I told you. Their job is to silence the gene expression. So that we call as a target. Suppose there is one microRNA whose target is oncogene. What will be the function of the microRNA? It will be a tumor suppressor. Suppose the target is a tumor suppressor. What will be the microRNA? It will be an oncogene. This is why we are interested in microRNAs. Because if the targets happen to be oncogenes, then that microRNA will be predicted to act as a tumor suppressor. And if the targets happen to be tumor suppressors, that microRNA will. So you have two types of microRNAs, oncogenic microRNAs, tumor suppressor microRNAs. Normally what happens is the oncogenic microRNAs are overexpressed in cancer cells as opposed to normal cells. Tumor suppressor microRNAs are underexpressed in cancer cells as opposed to normal cells, which means normal cells will have more RNAs, cancer cells will have less microRNAs, then it is a tumor suppressor microRNA. Cancer cells have higher microRNA and normal cells have low, then it is oncogenic microRNA. So this is what people have found out. Now, can we use that in therapy? That is the next question. Yes. There are only, see, this is a very important thing. What happens is, when the microRNA expression goes up, because it is a silencer, its target gene will go down. Its expression will go down. Correct? So whenever microRNA expression goes up, its target will go down because it's a gene silencer. Now this is a very important thing, because if you take a tissue, in a tissue, if the target happens to be an oncogene, microRNA target happens to be an oncogene, then what will be the role of the microRNA? It will act as a tumor suppressor. Simple. So you have only two things here: tumor suppressor microRNAs, oncogenic microRNAs. People give all fancy names, OncoMIRS, tumor suppressor microRNAs, like that they will give name. So what will be the use for therapy? How will you do it? You cannot introduce oncogenic microRNAs, right? You don't want to introduce. So you want to suppress that if you know that they are there in the cancer cells. So accordingly, there is this therapy also makes use of that. Just 1 minute. Forgetting that I should, yeah. I will not go into that. Yeah. So there are only two therapies. One is called microRNA inhibition therapy. If you know that the microRNA is an oncogene, then you inhibit that. That will be a therapy. If you know the microRNA is a tumor suppressor, then you want to introduce such microRNA, because that's called replacement therapy. So these are the two therapies that people usually use. I will not go into the details. I will go to the how much time? 15 minutes? Yeah. Because I have to show my paper publication and research work also. If I go on like this, it will. Okay. So this is what people have done. People have used many of these methodologies either to suppress the microRNAs or to introduce the microRNAs. So they have used it. Interestingly, in microRNA field, it has not come into the market yet. A lot of research is going on. Of course, some have come, but ultimately some of them have failed also. But in the area of cancer I am talking about. But then people are working on that, because this is one of the important fields where you may be able to get some guidance for cancer treatment. That's what they are looking at. So the people are working on many ways by which, for example, you may introduce certain oligos to inhibit the particular microRNA if it is a oncogenic microRNA. If it is a tumor suppressor microRNA, then you want to introduce that. So like that, there are many methods that people have started introducing. Let me see what can not just come. Okay. So they have used anti-RNA oligonucleotides, modified oligonucleotides. Then there are many methods. RNAs can act as tumor suppressors or even oncogenic microRNAs. So depending on that, sometimes what happens is there is a confusion because even in the same breast cancer cells, certain microRNAs, they can act as tumor suppressors, or in some other tissue, they may act as an oncogene. So lot of studies need to be done before we can take it appropriately to the clinic. That is one problem for this. Let me see. Yes. Lot of companies are interested. These are the companies which are interested in making therapeutics on microRNAs. Now, so by virtue of having multiple targets, a microRNA can have variable effects on oncogenesis cancer. Therefore, the goal is to fully clarify aspect before clinical application can be taken into. You change it, huh? Okay. We published this paper, what is called MIR 5096 mediated regulation of SLC7 induction of ferrototic cell death. I will explain that next. Probably we'll skip this next. Yeah. This particular molecule, which is called SLC7A11, it is overexpressed in certain cancers, and such cancers acquire radio resistance and chemo resistance. It's difficult to treat them. Next. Yeah. This is the molecule, what is called SLC7A11. We worked on that. It's a cysteine glutamate antipotent that exchanges intracellular glutamate for extracellular cysteine. It has a critical role in the generation of glutathione and the production of cells in oxidative stress. I will explain it in simple terms soon. See, what happens is this particular molecule called SLC7A11, it is upregulated in certain cancers like lung cancer, breast cancer, glioblastoma, ovarian cancer, and so on. Next. Pharmacological and genetic inhibition of this SLC7A11 can induce what is called ferrototic cell death and can enhance the cisplatin cytotoxicity. Some other people have shown that. You may be wondering what is this ferroptosis. You have heard of apoptosis. This is also one form of cell death. So I will explain that next. Ferroptosis is one form of cell death that we were kind of interested in. This is an iron dependent reactive oxygen species accumulation is happening there. And next. What happens is cancer cells are generally under hypoxic stress and are more susceptible to lipid reactive oxygen species induced what is called ferroptosis, something like cell death. But in this case, you have some iron related molecules involved in that. That is called ferroptosis. It requires glutaminolysis, a metabolic pathway, highly active in cancer tissues, and it is also essential for this metabolic pathway is essential for cancer growth, and hence it can be triggered as a ultimately as a therapeutic option. Ferroptosis, effector of cell death, is actually lipid reactive oxygen species. I hope you understand that. It's a little bit difficult topic, but go ahead. I told you about this SLC7A11, right? This is present in the membrane. Extracellular high concentration of is mediated by, you can inhibit that by extracellular glutamate and certain small molecules like erastin, sorafenib, and so on. Next. Can you go to next one? Yes, this I have already told what are microRNAs. That is okay. Yeah. Because the problem here is because of SLC7A11, which is a molecule present in the membrane, let us take, which is a receptor like molecule, which can be targeted, because this is actually providing resistance for therapy, because it's not allowing the cells to die. So ultimately what is aimed in cancer cells is generally to induce cell death, right? You want to kill the cancer cells specifically, right? So ferroptosis is also one kind of mechanism by which you kill the cancer cells. So that is the idea. That is the idea. So one of the microRNAs is underexpressed in human breast cancer cells and induces apoptosis by targeting SLC7A11. This is one thing. And similarly, we found these are some our background why we started this work. Next. This is about the binding site. Yeah, next. Okay. We ended up with another one microRNA called microRNA 5096, because you see this molecule SLC7A11 is the one that is conferring resistance. If you can put down that by attacking with a microRNA, then it will be easier to treat that condition. That was the idea. So the microRNA 5096 is the one that we found could be useful for this kind of work. It has 832 target mRNAs harboring totally about 997 binding sites, and 984 are localized in the 3' UTRs, because 3' untranslated regions are actually the sites for the microRNAs. That is why we were kind of interested in this. Next. So, no, go to that hypothesis. So, we thought that SLC7A11 might be targeted by this particular microRNA, because the UTR of that was matching with this, the sequence was matching. So, we thought this is a prediction, We have to do experiments to prove that, because as you know, once you have the complementary sequence, then such a molecule will be down-regulated. So, that was the idea. I know I am not explaining it the same way I have explained the other one, but since it is a little bit complicated. So, we predicted the microRNA 5096 might target SLC7A11 based on the sequences. Just now I told you that we have given the students also some targets and all that. So, in this manner we are able to predict, because you know the sequence of the microRNA, you know the target sequence. If they are complementary, then in all possibility such a molecule will be down-regulated by this microRNA. So, that was the idea. Next. But then we have to check it out. To experimentally validate if novel microRNA, what is called 5096, potentially targets SLC7A11. That is because we are able to find by bioinformatic means that SLC7A11 3' UTR contains the binding sites for this microRNA. So, we thought that it might be down-regulated by microRNA 5096. So, that was the idea. But then we don't know in what kind of cell death, because you see cell death, people have several forms of cell death. All of you must have heard about apoptosis, necrosis, paraptosis. One of them is ferroptosis. Ferroptosis is also a cell death one, but depends on iron. Ferro means iron. So ferroptosis is a form of cell death that requires iron related things. So that is that you take it simply like that. So this is one form of cell death. We became interested in that because of this particular work. Next. So one thing is, we have done some survival analysis. This particular SLC7A11 we took, and whenever this is present, the survival happens to be more. It seems to be anti-apoptotic or survival kind of molecule. So we thought, whenever you find something anti-apoptotic or something that provides survival, you want a molecule that can down-regulate that. Then you will be, that will be one way by which you can treat the cancer cells. If that happens to be an important molecule for the growth of the cancer cells, that is the criterion. That is the criterion. Here SLC7A11 is like that. That is why we took some breast cancer, this is from TCGA database, the cancer gene atlas, that's called TCGA. There is a database from which you will be able to identify certain markers, and you can see, if you think two things are opposite, you can see their expression database and find out whether whatever you are hypothesizing will work or not. So you will be able to deduce that. That's how we have come to this SLC7A11, and we found that in tumor cells this is overexpressed, adjacent normal cells it is low. So that means, in other words, SLC7A11 is a survival protein. So it is anti-apoptotic. Apoptotic, all of you know. Apoptosis means cell death. This is anti-apoptotic. So you want to suppress the anti-apoptotic molecule so that the cells will be killed. But this is not apoptosis. Initially we thought like that. Because it is anti-apoptotic, we thought that this will apoptosis. What I will tell you in brief, because there is no time. What we thought was, this microRNA 5096 we thought will induce apoptosis, but that was not inducing apoptosis. So we went ahead to find out whether there are any other mechanisms. For example, we know about apoptosis, necrosis, necroptosis, and ferroptosis. It so happened that we ended up with this particular microRNA inducing ferroptosis. Ferroptosis is one form of cell death that depends on iron. Ferro means iron. So ferroptosis is iron mediated cell death. You can take it like that. That particular cell death was induced by this particular microRNA. This was the first time a microRNA was shown to induce ferroptosis actually. We wanted to publish in Nature and like that. Unfortunately, fundings were not coming. I was applying for DBT and we didn't get the funds on time, and there were some issues. One student who started to work suddenly left the project, and I had to find another student and do. In the meantime, somebody else published. That is how we ended up publishing this ultimately in Cancer Letters, which is not a bad journal, but by that time other people also have published. That is one of the difficulties. You cannot speed up things, although it's a very novel work, that the microRNA inducing ferroptosis at that point of time was not known before. So this is what we have shown. I will quickly go through the results. Can you move to the next slide? Yeah. So we saw that higher expression of SLC7A11 contributes to poor patient survival. That from the TCGA database, the cancer gene atlas, we found out that. Next. Why I also did not tell you in detail, this SLC7A11 is a molecule that provides survival. And in that 3' UTR, we found the binding site for this MIR 5096. That is why this is theory. from bioinformatic analysis. That is why we took that microRNA and tried to see whether it can downregulate that. That is the story. And finally ended up showing that it acts through ferroptosis mechanism. That is what we have shown, and we published it also. Yes, go ahead, and I will stop here almost. Next. So no, go to the red ones, because that tells. We found out that microRNA 5096 may act as a tumor suppressor microRNA in breast cancer cells, and SLC7 is overexpressed in breast cancer cell lines and can act as an oncogene in breast cancer cell line. That is why this finding is important, because we are showing a microRNA can suppress the oncogene. See, the interest in microRNAs in cancer biology is because if you take a microRNA and its target is an oncogene, what will be the microRNA? MicroRNA will be a tumor suppressor. If the target is a tumor suppressor, the microRNA will act as an oncogene. So the microRNA is a double-edged sword. It can act as an oncogene, it can act as a tumor suppressor gene. Accordingly, you can play with that. So there are only two therapies involving microRNAs. One is, if you know that microRNA is an oncogene, then you employ what is called microRNA inhibition therapy. If the microRNA is a tumor suppressor, you are going to introduce that. Usually such microRNAs are low in tumor tissues. So it is called microRNA replacement therapy, because it is not there, you are replacing it. It is in low quantities, you are replacing it. So there are only two types of therapies. MicroRNA inhibition therapy, if the microRNA happens to be an oncogene. If the microRNA happens to be a tumor suppressor gene, then you introduce that. Here, SLC7A11 is anti-apoptotic or survival protein. So we introduce the microRNA to suppress it. We realized that it is not through regular apoptosis. That's what we thought initially. But it is through ferroptosis. That is what we found. Next. Yes. Next. Next also. Go ahead quickly. Yes. So we showed that it can post-transcriptionally regulate SLC7A11, which means that the mRNA level, it can downregulate SLC7A11. Next. We also found the binding sites for that, and it is matching. So we are able to show that this particular microRNA can silence the SLC7A11. Next. And we have done what are known as luciferase assays. These assays will tell us that whether this SLC7A11 is indeed a target for this microRNA, because you are attaching a luciferase, and if we introduce the microRNA, the SLC7A11 will go down, its expression will go down. That is measured by luciferase assays. So we will know that the expression goes down. Next. Yeah, we showed that down regulates SLC7 by targeting its 3' UTR. You remember we showed the sequence similarity. That is by bioinformatics. We saw the sequences were complementary. So we want, just by showing it, it doesn't work. I told you in the beginning, my experience was that it doesn't work that way. In vitro and in vivo may differ. In this case, it worked. Whenever it works, then you publish a paper showing that we first did bioinformatic analysis and then experiment, and it worked. That's how people are doing actually. Yes. Next. So we looked at the ectopically expressed microRNA 5096 on proliferation, and we have looked at whether the cells are dying. Next. Inhibits cell proliferation and increases cell death. Yes, as we expected, this microRNA is able to kill cells. Next. We have done several other experiments. I will not go into the details. Can you go quickly? I will just read out this. Apoptosis does not contribute because some of the apoptotic genes were not altered. So we know that it is not through apoptosis. Next. There is also another phenomenon called necroptosis. There are so many terms. Necroptosis, paraptosis, apoptosis, all these things are there. each one has a gene, and so you look at these genes whether they are altered or not. You look at it and then rule out one by one and finally end up with ferroptosis. Next. Next. That's what we have done, necroptosis, and it is not necroptosis. So we have, again interestingly, we have used two chemicals, ferrostatin and erastin. Erastin can induce ferroptosis. Ferrostatin can block ferroptosis. So we use these inhibitors. Next. Next. This just go next. I want to see that. So by overexpressing the microRNA, we have shown that it can produce reactive oxygen species, which is the real killer of cancer cell. So we found out that this microRNA is actually killing the cells by enhancing the production of reactive oxygen species, which can kill the cells. Next. So we, because ferroptosis is something to do with iron, we looked at iron accumulation and oxidation of glutathione, but decreases the mitochondrial membrane potential. Next. Sorry about skipping many slides, but next. Next. We have done some electron microscopic studies also, and overexpression leads to mitochondria shrinkage, which is typical in ferroptosis. Next. Next also. We will finish with next. If you restore the SLC7A11, it can reverse. See, if I say that SLC7A11 is downregulated by this microRNA, you introduce SLC7A11 and see whether it is reversing. Always these scientific experiments are always done like that. So we have done that, and we can look at survival, reactive oxygen species, lipid peroxides, ion accumulation, glutathione, hydroxyl radicals, and membrane potential, and so on. Next. So we also looked at, can you go back? Yeah, go down, it's okay, it's okay, it's okay. There is a sum up I must have written. Next. Yes. So we found that it can inhibit colony formation, transwell migration, the migration is inhibited, then invasion is inhibited by this microRNA. So in short, we have shown this microRNA acts as a tumor suppressor microRNA. Next. Next. Ectopic expression of the microRNA alters the expression of EMT. We have also shown that it can inhibit what is called epithelial mesenchymal transition, EMT. EMT is an aggressive form. So it can aggressively transfer the cancer cells into migration. So we have shown that it can also inhibit that. Next. Finally, people don't accept if you do anything in cell lines, I told you. So we wanted to take some animal system. So we didn't have these nude mice and all that. Finally, we took zebrafish model, and where we have shown that it can inhibit metastatic potential. This is a breast cancer cell line. Next. So we have shown that this microRNA, finally I will sum up. We have shown that this microRNA is acting as a tumor suppressor microRNA, and it is acting through the suppression of SLC7A11 3' UTR, because that is what a microRNA does. What microRNA is doing? It can silence the gene expression by binding to the 3' untranslated region of the messenger RNA, thereby inhibiting the translation. Ultimately, translation is inhibited. Okay, that is what we are. What is the protein that is inhibited? SLC7A11. What is its function? It is actually a survival protein. We have shown that this novel microRNA actually suppresses the expression of SLC7A11, which is shown to provide resistance to cancer cells. So that's what we have done. We have done that. We have next to the final one. Next. So these are the conclusions. We have shown that it can regulate SLC7A11. It can induce ferroptosis. Ferroptosis is a form of cell death that requires iron. And if you ectopically express, it can show tumor suppressive properties, and mediated effects are partially restored by reintroduction of SLC7A11. If you say that SLC7A11 resist, can you reverse the effect? So, introducing it, we reverse the effect. Next. Next. Future directions, it's okay. It came in some newspaper, they have said researchers zero in special type of RNA and so on. So I'll stop here. Sorry that I took so much time. Anyway, it was nice interacting with you people. Once again, thank the organizers. Yes, let them ask.
Hello. Yeah.
Yes. Yes.
I have a couple of questions.
Yeah.
One is, do you have any chemical that can increase the microRNA levels in human or any human cells?
That is a good question. See, I showed you, see, after all, these are gene products. So, if you can stimulate that, it should be possible.
Okay. Second thing is, since you know SLC7811 has potential to survive that particular cancer cells.
Yeah.
As you know the 3' end of it, UTR region, can you synthesize an mRNA specific to that?
MicroRNA,
so that you can target
not mRNA, miRNA.
mRNA, sorry,
Correct.
mRNA.
Yes.
And then you can target.
They are called microRNA mimics.
Yeah.
The problem is, if you put a raw nucleotide into the cell, that will be destroyed by.
No, I am just putting a.
No, correct.
No.
I am telling you what is happening. So what will happen is, they will attach some nucleotides which cannot be degraded by nucleases.
Yeah, okay.
So in that manner, that is called modified oligonucleotides. So if you can introduce modified oligonucleotides, you should be able to. And they will give tandem sequences so that you will have more of this microRNA attacking that, like a sponge. That also we call it a sponge, and we have used that sponge methodology also. It works for another microRNA,
okay.
microRNA 155. We have done that. It's possible. But the problem is same. Like when you say this liposomal and all that, you remember the famous quote said. He was the pioneer in the antiviral vectors and all that. He said there are 3 problems in gene therapy. What are the 3 problems? Delivery, delivery, delivery. That's what he said. That is, how to deliver that into a particular area is one of the typical problems. Here the other problem is that nucleases can destroy some of the oligos. So you have to modify them and then introduce. And in the there are some side effects, and they stopped in the case of microRNA 34. So still it has not gone to the human therapy in cancer, but in other diseases the microRNA is able. So people are now using for detecting or for diagnostic and prognostic purposes. That is very useful. Yeah.
So you said that there are different types of apoptosis, sir, like ferroptosis.
I don't need any. Yeah.
In that, only ferroptosis that is caused by the cell death is caused by because of iron, no, sir. Like that, other magnesium, like other ions, like magnesium, like phosphorus, like that can cause ferroptosis, sir. Is there any study, sir?
It's not like that. In this case, they found, but different forms of cell death people have found, necrosis, and all that. In this particular case, iron-related things are required.
Yes, sir. Like that, potassium, like that kinds of, that types of ions can cause apoptosis, sir?
We don't have anything specific like that.
But we can study about this.
Yes, yes, one can study. It is just that they upon this.
Oh.
It's not that every ion will be important for cell death, you know. It's not like that.
Good afternoon, sir. Sir, you have talked about the repressing effects of mRNA. So, what is, can they upregulate any gene expression?
The basic thing is, if you suppress a suppressor, you
like I'm
you are suppressing a tumor suppressor, you will be promoting tumor growth. Inhibit an inhibitor, you will activate. Correct?
Yes. Like, is there any specific gene will it upregulate, sir? Like, I'm not in cancer, sir.
Yes. The problem is, this is a multi-gene thing. I told you, one microRNA can target 500 genes. Similarly, one gene can be attacked by 150 microRNAs. Multi. Each side it is multi. Target also can have multiple microRNAs. So the net effect is what is happening in the tissue or in the cells. So it is complicated. It's not as simple as that. That is the idea. We have to see what happens and then come to some conclusion. We cannot just like that conclude. That is why these in vitro predictions fail. Because you predict through bioinformatics, okay, this best microRNA, best target, but in vivo there are many other factors which ultimately defeat what you predict. Yes. Yes. That's okay. Yeah.
Hi. Thank you. Each microRNA controls 500, 700 different genes. How would you create this selectivity or specificity to target a certain mRNA rather than to target many others?
Yeah, that's a very good question. For that only, the siRNAs are there. You can do that. Otherwise, in microRNAs, if you want to do that, usually what happens is in microRNAs, in animal systems, generally, there is only a partial complementarity to the binding sites. Whereas in plants, there is 100% complementarity. There it will completely downregulate. Yes. In plants, such a system is there. But the problem is, such kind of 3' UTR sequences may be present in some other genes. If they are present, then the selectivity is lost. Yeah, I know. The problem is, if you have that, then you don't have much ways to do that except to adapt siRNA kind of approach. Find out a region where there is no real similarity, but there is a complementarity to the particular siRNA, or you design on your own and then do it. MicroRNA is also, you design in such a way that the complementary sequence does not exist in other things. Then you can do it.
I mean, the main problem is,
yeah.
No matter how big or how small you do it, there is definite sequence similarity. If one missing, it doesn't matter, it's going to target.
Correct. At least partial sequence, this thing will be there.
microRNA looks at only partial.
Yeah.
So when it's looking for you have 21 base pair. Even 20, you'll find so many 20 sequences. Because if you look at the human genome,
Yeah, I will answer that question. It is complicated, but cancer is also complicated. See, the problem is, in cancer, it is not one gene defect. So, multi-gene. And microRNAs are born because they affect multi-genes. So, in that sense, it is useful. But what kind of multi-genes they affect will ultimately determine whether they are useful or not. That is the problem. That is always there. See, sometimes what happens is, if the cancer cells are addicted to one particular gene, for example, HER2/neu, it is only in 20% of the patients that this particular gene is overexpressed and the cells are addicted to this gene. In such cases, even if you affect this gene, you can cure cancer, or you can at least treat cancer to a satisfactory level. If that is not the situation, then you need to have multi-gene attacking. That is the idea. So, basically, a fundamental knowledge and a thorough evaluation of the system is essential before you attempt therapy. If you do not do that, one day it will back cross on you, it will come across, it will come back to you as a boomerang. That is what happened in many therapies. Many therapies have been withdrawn because people have not studied it thoroughly. This is a perennial problem, it is not a simple problem like that. But it can be taken as an advantage, because here is a tool which can affect multi-gene, and cancer is a multi-gene abnormality. So, it is an ideal tool in that sense. But there must be a match there. The multi-genes affected should also be affected by this microRNA. Yeah.
Because cancer multi-gene is correct, but mRNA will not attack only those multi-genes. So it might attack many multiple other systems.
Correct. So that, but in some cases it has worked. No, it is very interesting, and it is true also. Where it works, you take. Where it doesn't work, you don't continue. That is it. Or try to see whether you can modify that. That's what you can do. What can you do otherwise? Yeah.
Sir, there is a question from the online participant. Can I read it out? The name is Ranjana, student of physiotherapy, second year, from Muhammad Sadiq Aj College of Physiotherapy, Chennai. The question is, sir, like we have found vaccines for rRNA viruses like hepatitis C, and why can't we find vaccines for diseases like Ebola, HIV, Zika virus? What could be the major reasons? It has been years for HIV, etc.
No, I am not a vaccine specialist. You don't ask questions that are totally unrelated, you don't ask.
Yes, just general questions, so I just thought I yes, okay. Okay. So there is also another one. Why DNA, same type of question. Why DNA viruses vaccines are more stable and curable, and why not RNA, sir? Same question. Yes, sir. Yeah. So, yeah, you can ask.
Vaccine type of questions, I am not a vaccinologist or anything. Yeah.
Good afternoon, sir. Sir, you answered to one question about mRNA mimics, sir. So, during your research...
I would have said miRNA mimics.
miRNA mimics, sir. Sorry, sir. So, during your research, did you find any miRNA mimics interrupting your work, sir? If so, what is it, sir?
We have not done it that way. Please sit down. In one of the slides I might have shown, we worked with one microRNA 155, where we have put sequences of the microRNA side by side and cloned them. So that means in one construct that we have, there will be, because it's a small sequence, you put them side by side, 3, 4 of them, and then put them onto cells. What will happen? The complementary sequence they will take out. So that means in the kitchen, when we have some water droplets and put a sponge absorbs the water. Here it is pretty much the same. Those microRNAs can be depleted because you have the complementary sequence here. So they can be depleted. That is known as microRNA depletion. You can do that. That is a sponge therapy, you can say. Yeah, sponge construct. We have used that and it worked in some cells.
Good afternoon, sir.
In oral cancer cells, we have done that. Yeah.
I'm B. Sudha from first MSc Biochemistry. And my question is, at last you have said that the gene or protein SLC7A11, which was inhibited by the miRNA. So at the end, it also get reversed by adding the SLC7A11. Do you think it is a disadvantage for using this miRNA as a therapeutic agent because it reversed its action? No matter what, it have to kill the cancer cells. But adding the SLC7A11, it again reversed from its action. So how can we use this as a therapeutic agent, sir?
That is to demonstrate two people are there. If they are having equal power, suddenly you are increasing the power of one person, he is going to win, right? It's something like that. SLC7A11 and the microRNA are two opposing things. It depends on what you increase. If you increase this, naturally he is going to win. So the relative, in this case, the relative expression levels will determine that. So if one is going up, you have to increase the other. That is.
Thank you, sir.
Cancer incidence and mortality rates stretch significantly across different regions and countries. Thank you for an informative presentation that has significantly enhanced our understanding of breast cancer therapeutics. Thank you, sir, for sharing some important updates on life science work. Announcements. Of poster presentation, kindly assemble at GRD Auditorium foyer. The judge will evaluate on participants of poster presentation, kindly assemble at GRD Auditorium foyer. The judge will evaluate on 2 p.m. Fuel afternoon. Lunch time is here. Lunch for the registered participant is arranged behind the auditorium. Kindly have it and assemble here at